Let us go ahead and get started. I'll be honest, I'm a little surprised by the lack of people who have shown up so far already. I've just started grading the exams, but what I've seen in the lab exams so far is looking pretty good. So all I can figure is maybe everybody's really depressed that Claire is going to be gone after the next episode of The Bachelorette. So maybe everybody's still a little hungover from that. All right, let's go ahead and talk about our game plan. Today is Thursday, October 29th. And we are on to the fourth section of this class, and that is going to be two systems. So we are going to quickly through the digestive system. We will go equally quickly through the respiratory and urinary system. We had the advantage of just one organ system for the last one, but it was a very quick turnaround. We'll have a little bit more time, but we're going to be cramming two organ systems into that. And so that is going to be a lot of work. There's some similarities to them. Both of them have 2 unit reviews you're going to be responsible for. Both of them have a Physio X you're going to be responsible for. And both of them have an additional lab that you'll be doing as well. So let's quickly talk game plan. Tuesday, get up early, get out, vote, and be back here in time for class at noon. If you haven't voted already, please make sure you get out and do that. And your Unit 22 review is due, so you can do that while you're waiting in the long lines. Your Thursday unit review 23 is due, as is a home lab. This home lab is a fun activity where you've got the opportunity to do some of these things at home, but I want to talk about the rules of this clearly first. So I'll make sure I've got this at the beginning of the recording so that everybody is fully clear and understands what they are doing. If you have any type of respiratory issues, if you have asthma, if you have any other type of COPD, any other type of respiratory issues or cold or asthma, or even the seasonal flu or anything like that, do not do these activities. All right, do not do these activities alone. Some of the breathing activities that you'll be doing in this can make you lightheaded. So make sure you are seated when you're doing this, and make sure there is somebody observing you when you do this. If you have respiratory issues and there's somebody in the house that is comfortable doing these activities, then you may observe them do them and follow the rules for that. Have them do these activities, but do not you or anybody else do these activities by themselves. Make sure you have somebody observing them. Make sure they're seated while they're doing all these activities. Also, while some of them is as simple as holding your breath and timing yourself, there is the opportunity to make a pH indicator, and the rules are on there. If you do not have the resources necessary, or not comfortable going to the store to buy those resources, or can't get them, dropped off to you by Instacart or anything like that, that is okay. However, if you cannot complete the activity, then in reality, then I want you doing it as a thought experiment. I want you writing out what you think would happen and why it would happen. I do not want any blank answers for any of these. So it's okay if you can't do any of the activities, but if you cannot do any of the activities, then I want you to write out what you think would happen and why you think that that would happen. So there's no reason you can't do this as a thought experiment if you're not comfortable or capable of doing any of these activities safely any other way. All right, so this is an important assignment. It is something that I expect you to do. But again, if for any reason you're not comfortable doing any of the activities, you are not required to do that. In fact, you are prohibited from doing them. But instead, just write them out as a thought experiment as to what you think would happen. All right, so hopefully that is crystal clear on that. Again, your Physio X is due on the 10th. That is 3 activities. So that means 3 lab reports. You guys have done a lot of Physio X's, so I think you've got that system down now. Thursday, the 12th, you have your unit review. But again, what is correctness? If you're holding your breath and you can only hold your breath for 30 seconds and somebody else holds their breath for 45 seconds, and then which one is correct? So it isn't so much that I'm grading it for correctness, but I'm definitely grading it to make sure that you have either done the activities or thought about the activities. So it is being graded for completeness. I want you putting time and effort into these, right? On some of the reviews, I see answers that are 2 or 3, words long on something where it says, is it A or B and describe it? And as long as you're writing for most of them, I tend to look the other way on that. I will be more strict about this one. It is okay not to do the activities, but I want you showing me that you put time and effort into thinking about those breathing activities. All right. Then we move on to the urinary system. And again, 2 unit reviews. Unit review 25 is due on the 12th. Unit review 26 is due on the 17th. You have another Physio X, exercise 9, due on the 17th, which has 6 activities that you are going to be doing. And also due on the 12th, you have another Labster. Again, remember, 2 weeks, as well, 3 lectures. So basically 2.5, a week and a half to learn this material. Absolutely. So I think it's 1, 2, 3.5 weeks total for this section. Yes. So again, get started early. This weekend is one of your weekends to use to start learning this material and get ahead on this material. The Labster, remember, you must complete it in entirety, and to get full credit for it, you must get at least 80% correct. Remember, there's been a couple people who are not completing it to 80% correct. You can do it a second time and improve your grade. So improve your grade, make sure you get full credit for doing that. If you don't get it to 80% correct, then you just get whatever partial credit you got for what you did get correct. So make sure you do that as well. Again, as Ashley pointed out, we do have a quick turnaround with this section, and we also have one of those dreaded Thursday exams. What that means is that you're being presented with new material on Tuesday and being graded on it 2 days later. However, just because I'm presenting the information to you on Tuesday doesn't mean that you shouldn't be looking at it ahead of time. Again, you have the weekend before that. So I encourage you to look at that material ahead. You have the lecture notes, you know exactly the things that we're going to be talking about. So start this weekend, get ahead, start that weekend, get ahead, stay ahead of this material so that you can be successful on that Thursday exam. All right. Questions on that? Excellent. We are rapidly approaching.
May I ask?
Oh, go ahead, please.
About the time that you give for exam, so you regulate the time. You don't usually give like 150, but sometimes you give less, as this time you gave just 120. I mean 120 minutes for the lecture exam.
I give what I think is an appropriate amount of time based on the amount of work that is being asked for you. The normal exams up to this point have had closer to 30 to 35 multiple choice questions on them. They've all had 8 essay questions on them. This one had 7 essay questions. This one only had 25 multiple choice questions. So it was a smaller amount of information, which is why it was a smaller time. It's the same thing with the lab exams. I give the time of the lab exams based on the number of questions. So I don't have a set time that we have for all of these exams. It's based on the expectation of how much work should it be and what's an appropriate amount of additional time to add to that.
Okay, thank you.
Yeah, but now again, without even having thought about the exam yet for this section, the fact that we have 2 organ systems we're going to be working on means there's more likely that there's going to be 8 or 9 essay questions. It means there's going to be more multiple choice questions. So my guess is, yeah, it'll be closer this time to 2, 2.25, 2.5, depending on how many questions end up on this exam. But yeah, I always try to gauge it based on the length of the exam. Yes, absolutely. You are responsible for all the material we do in this class. If I give you a graded assignment like the Physio X or a review or a Labster, is it possible that that is testable material? Absolutely. And so, yes, some of you likely got a question about the labs that you were doing in the Physio X. Remember, especially in digestion has a lot of wet labs that we normally do in the classroom. Unfortunately, we were reliant on having to do that virtually. So I don't think that there should have been a surprise that there was something related to that. And same thing here. Could one of these, you're getting 2 Physio X activities, a home lab activity, and a Labster. Do you think the labs are important in this section? Do you think that there could be essay questions on the lab? Absolutely. All the material we cover in the class is the material that you are responsible for on the exams in all the formats that we do it. Sometimes, occasionally, the review questions from the unit reviews, the check your understanding parts are occasionally essay questions in some of in the banks for those. So some of you guys have had some of those as well. Yeah, all the material you do in this class is something that you are responsible for. Well, why do you think I make you do the activities? Just for the heck of it, because it's fun? No, it's the point is to learn this material. So it hopefully won't be a surprise in the future. All right. Any other questions? All right, look, think back. We did blood typing activities in lab, and there were blood typing activities on your exam. So from the first exam, lab things you've been doing are things that are on the exam. Yep, you're responsible for everything we do. All right, any other questions? All right, so again, this is not an unfamiliar format. This is something we're basically going to do with both of these two sections. As you see, we have 3 days for each of these units, each of these organ systems. And so for both of them, it's going to go the same way. On day one, we are going to hit the anatomy hard, make sure we understand all of the structures that we are responsible for. And then, great question, I'll answer that in a second. Let me just finish this. So the anatomy on day one, and then the other 2 days we'll focus on the physiology of these systems. Like I said, I've just started grading. Usually it takes me about a week. Obviously I did have done some grading up till now. I will continue to grade tomorrow and Friday and Saturday. And as always, as soon as I get the grades done, I will post the grades. And then again, on the day on Tuesday, usually I try to get it done by a week. And again, knock on wood, I don't see any reason why that shouldn't happen this time, as always. But again, you know, it is 2020, but I will do my best to get them done and back to you by before Tuesday, or at least the grades posted on Tuesday. And then like before Tuesday after class, I will release the exams so that you can view them for 24 hours. And then I'll close that down on Wednesday again. So yes, that is my expectation of what will happen with these exams as well. So far, I'm still on schedule. And like I said, knock on wood, with this weekend coming up, I should be able to get a fair amount of grading done, if not all of it. All right. Any other questions on the game plan? Thank you. And any other questions on anything else? I appreciate that. All right, then let us dive in. So, like I said, today we are going to focus mostly on the respiratory anatomy, but we need to talk a little bit about the respiratory system and what it actually means when we talk about respiration. Because often when we think of respiration, we just think of this, right? That is the mechanical events that are associated with what we call breathing, or what we call our pulmonary ventilation, the movement of air into and out of our lungs and through the respiratory structures. However, as we know, the whole reason for getting that air into our lungs is so that gas exchange can take place. And let's talk some specifics about what gas exchange means. When we talk about gas exchange, there are a couple ways we can think of this. We can think of it in terms of the structures where the exchange takes place. What would the structures in our external gas exchange be?
Oxygen.
Okay, oxygen is one of the gases, but excellent. All right, so lungs is getting us closer to it, but what specific structure? There you go. So excellent. I like that. That's a good start. Lungs and blood vessels, or blood capillaries. But I think we can be even more specific than that. What specific structure in the lungs where the gas exchange takes place? I know we haven't really talked about the anatomy yet, but I think most of us know it. Excellent, Vanessa's got it. The alveoli, or alveolus, will go singular. So an alveolus in the lungs. So let's put lungs in parentheses here, because specifically external gas exchange takes place in the alveolus. And I think she also pointed out in our pulmonary capillaries. Those are the two structures where our external gas exchange takes place. You guys are doing a great job of naming organs in the respiratory system, but again, that's not where we're looking for. Gas exchange doesn't occur in all of those. It only occurs in one location, and that is in the alveoli. Oh, and I said alveolus singular, so I should be singular with my capillary as well. But as Oksana also pointed out, there are also mediums for that exchange to take place, right? I heard one of those mediums is air. And of course, that is the medium, the substance that is in the lungs. And what is it in the capillary?
CO2.
Well, okay, once again, those are the gases that are being exchanged, but the actual medium, the substance where the exchange is taking place is between the air and, there we go, the blood. So when we talk about exchange of gases, in specifically external gas exchange, we can describe it two ways. We can describe it in terms of structures, or we can describe it in terms of mediums. External gas exchange takes place between the alveoli of the lungs and the pulmonary capillaries. Or we can say it takes place between the air and the blood. Now, as you guys have already pointed out, when we talk about the gases that are involved in this, what are the gases that are involved in this?
Now it's oxygen and dioxide.
Excellent. One is indeed oxygen, and the other is carbon dioxide. And in our external gas exchange, which way does oxygen move?
In?
Well, but in what? In the air, in the blood, in the alveolus, in the pulmonary? Let's be more specific. Let's go structure to structure or medium to medium. Oxygen moves from what to what?
From air to blood. No?
Yeah, oxygen moves from the air into the blood, right? Or you're right, we could have said from the alveolus into the pulmonary capillary, and I'll abbreviate that in the interest of times. Excellent. No, great question, Ryan. Nitrogen does not play a prominent role in gas exchange. As we'll talk about later in the week or next week, while nitrogen is the dominant gas in the air that surrounds us, its role in gas exchange is very, very minimal because it basically doesn't dissolve well into water, which if you think about it is what blood is. Excellent. Which way does our carbon dioxide move?
From blood to air?
Yeah, there you go. From blood to the air, or again, we can talk of it in terms of from the pulmonary capillary into the alveolus. All right. Everybody get how the game is played? So when we talk about external gas exchange, this is what we are talking about. And let's cheat, although I don't know how easily I can do this. Maybe if I condense this all back down. All right, I need space, so sneak that up there and do that. That works. All right, excellent. Now, of course, anytime we have an external something, what else are we going to have?
Internal something.
Exactly, internal something. And so we need to have that internal gas exchange. And so let's do the exact same thing. What are the structures that internal gas exchange takes place between? All right, I like that. Blood and cells. So again, blood I think would be more of a medium. So what of course is going to contain that blood? There you go. I like that. Systemic capillaries. Right, well, I should be consistent with my, so a systemic capillary and the tissues, again, where the cells are located, because the blood doesn't go directly to the cells, it goes to the tissues. The interstitial fluid, which would be our medium, would then be indeed, would be blood. and the interstitial fluid. All right, that's after all, when we talked about our capillary exchange, when we talked about it, that's what we talked about it, between the blood of those systemic capillaries and the interstitial fluid of our tissues. Excellent. Of course, once again, what gases are involved in this internal gas exchange?
Oxygen and carbon dioxide.
There you go. Same two gases, but same directions. Which way does oxygen move in internal gas exchange? Excellent. In this case, it moves from the blood into the interstitial fluid. Remember, we want to go medium to medium or structure to structure. So it would be blood into the interstitial fluid. Or we could do systemic capillary into the tissue. So again, we don't want to mix our structures and our mediums when we're describing this. And so then which way does our carbon dioxide move? Excellent. From our tissues into the systemic capillary. And again, for consistency's sake, we will say from the interstitial fluid into the blood. There you go. And that is internal gas exchange, right? These are not necessarily new concepts, right? These are things that we've kind of talked about in the cardiovascular system and other places along those lines. And of course, we need to get from the pulmonary capillaries to the systemic capillaries, and of course back again. And that movement of the gases in the blood is what we call our gas transport. So when we talk about the importance of the respiratory system, these are the things that we are talking about. Of course, the whole point of this, the whole reason we do that is so that our cells can do a process called cellular respiration. Can someone remind me what cellular respiration is again? The chests of our little individual cells heaving as they move those gases into and out of them. Give you the answer is no. Why do... All right, excellent. Absolutely. Our cells use oxygen to produce ATP. If only some point way back in 430, we had learned the equation of why that oxygen was so important. Because what is it that that oxygen lets us do? How do we use oxygen to make ATP? Okay, but does oxygen really need to be used for just the splitting, glycolysis? There you go, excellent. So excellent. We need glucose, you're right, we need fuel. So oxygen plus our fuel, sugars, although it can be fats and in some cases even proteins. We use that oxygen to completely break down that glucose. Where does that take place again? Mitochondria. Excellent. So we use oxygen to completely break down the sugar. And as a result of that, we get lots of energy. And that energy with the electron transport chain, as we know, is used to make ATP. However, there are some waste products in this process. And what Krebs cycle or citric acid cycle, absolutely. What are the waste products of this process? There we go. Water and carbon dioxide. The whole reason you do is so that you can get the oxygen you need to the cells, and you can get rid of that carbon dioxide waste that the cells made as they're making their ATP. So the whole point of the respiratory system is so that every single cell of your body has the potential to be able to do this cellular respiration in their mitochondria, where they completely break down sugar by using oxygen to produce massive amounts of energy. Remember, one glucose, when completely broken down, can give us somewhere between 42 and 46 ATP. It is incredibly efficient. Like it's like that hybrid Leaf car that you have, or those are electric. So hybrid Prius or whatever that you have. That allows you to be much more efficient in your energy, but it gives off water and carbon dioxide as a waste product. All right. Now, I think we did most of this, but again, there's the pretty picture that goes along with this. Pulmonary respiration is what we call ventilation. What's the primary muscle involved in ventilation again? Diaphragm, excellent. All right, so we use that diaphragm, but as we also know, and we learned in 430, there are plenty of accessory muscles that we'll talk about that can assist in that process. We talked about external respiration already between the alveoli and the pulmonary capillaries. We did all that. We talked about internal respiration or gas exchange, gas diffusion. And that's the one other point that I should make about this as well. These are diffusion processes. What's so special about that word? No ATP. As vitally is used in this process. As vitally as important as it is to get oxygen to our cells and get gases out of our cells, we do not use any ATP to do that. All of the diffusion of the gases, all of that gas exchange is passive. It uses diffusion. So as we know, we're going to need a very thin membrane, which we already know we have in our capillary. It's that endothelium, that tunica intima, that single layer of simple squamous epithelial tissue that allows the rapid diffusion. And we'll see in the lungs, we have a very, very thin respiratory membrane and a massive surface area. We have massive capillaries, we have massive surface area to our lungs to facilitate that rapid gas exchange to take place. All right. And like I said, we have that cellular respiration that we talked about as well. All of these things are associated with our respiratory system. But how many of these four functions are actually the function of our respiratory system? Of these four things that are called respiration, how many of these are actually done by the respiratory system? One, which one? Two, which two? All right, ventilation, any others? One and two, there you go. These two, external respiration and pulmonary respiration, the mechanical movement of gas into and out of our lungs, and that gas exchange that takes place between the lungs and our pulmonary capillaries. These are the two that are the responsibility of our respiratory system. All right, excellent. So there you go. Those exactly. Those are going to be the two. While we'll talk about the other two, internal respiration, and again, I just mentioned cellular respiration, we are going to focus primarily on the functions and the processes of these two. So there you go. We did that. Perfect. Potentially, We will see that typically the two go hand in hand, absolutely. But as we will see, oxygen level and CO2 levels in the blood are independent. So is it possible to have blood that is high in oxygen and high in CO2? Yes. Is it possible to have blood that is low in oxygen and low CO2? Yes. However, under normal function is typically oxygen levels. Don't know who that is, don't care. Typically, when oxygen levels are higher, CO2 levels are lower, and vice versa. But we will actually, again, if you've learned one thing from me in the half a year, or for those of you a year and a half that you have taken of me of 430 and 431, I'm not a huge number, guys. Numbers are things that you can primarily look up. But as we also know, there are times when numbers are important. And there are going to be some important numbers we're going to learn. And those actual levels of oxygen and CO2 in the blood are going to be one of the numbers you will be responsible for learning. So we will talk about that, and you'll actually see the condition of the oxygen and how it changes, and the condition of the CO2 and how it changes. All right, excellent. So for the respiratory system, we are going to talk about the functions of that respiratory system and the organs. Their job obviously is to facilitate gas exchange, but as also is important, it's important not to just take a chunk of this Sacramento air and shove it into my lungs. So we need to not only move that air into my lungs, but we need to process that air along the way. We need to, I see your question, Ryan, let me finish this sentence and then I'll read it. We need to warm the air so that gas exchange happens more readily. As we know, temperature affects diffusion rate. We need to moisten the air because the gases, oxygen and CO2, diffuse more rapidly when they are dissolved in water. So by moistening the air, it's going to facilitate diffusion. And we need to clean all the pollutants that are out here in the Sacramento air that before it reaches that delicate membrane in our lungs. It's one of the reasons that we talked about in the past, how our nose is not a part of the digestive system, even though you could get milk from your nose into your stomach. The same way the mouth is not part of your respiratory system. While it is a passageway that air could use to get to your lungs, it doesn't filter and process the air the same way, so it is not considered part of the respiratory system. All right, let me read your question now. I promise you we will talk about that nitrogen toxicity that happens with divers. We will get to that. We need to talk some anatomy first before we can get to, as I've said many times in this in 430, chemistry is a completely stupid and pointless study. Nothing good has ever come from chemistry, but physics is a tremendous amount of fun. And we are going to spend a fair amount of time talking some physics. And in physics will help us to understand exactly what happens to those divers and the nitrogen in the blood. So we will absolutely positively talk about that. All right, so now to random question and all, a very relevant question, but we need to build to it first. All right, now, again, those are our primary functions of the respiratory system that meet our goal of allowing that oxygen into our cells and getting rid of the CO2. But our respiratory system serves other functions as well. Obviously, it provides us with our sense of smell, that chemical, recognition of chemicals dissolved in moisture. And remember, that takes place way up here along the superior border of the nasal cavity, and we'll talk about some of the anatomy of that in just a minute. But our respiratory system also plays a very important role of helping us to regulate the pH of our blood, which, as we know, of course, regulates the pH of our interstitial fluid, and therefore regulates the pH of our entire body. How is it that our respiratory system helps us to regulate our pH? Anybody know the trick to that? There's this mystical, magical chemical equation that takes place. When carbon dioxide comes in contact with water, oops, so I spelled water right. What do you get as a result of that? Does anybody know? An acid, absolutely. Carbonic acid is absolutely correct, but for right now, we'll just say an acid, absolutely. So the more CO2 your body is producing, the more acidic your fluids, the humors of your body can become. And therefore, the more important it is for us to expel that CO2. So when your body's more metabolically active, like when you're exercising, what happens to your respiratory rate?
Go up.
Yeah, it goes up, absolutely, to help to not just get oxygen into our blood, more oxygen for that more metabolic needs, but also to help us get rid of that CO2 as well. In fact, too much emphasis is spent on oxygen. As we'll see, CO2 in many cases is just as, if not more important than oxygen, especially in determining your breathing rate. What do you do when you don't get your way? Come on, hold your breath. I don't want to go to school. And your wife says, tough, you have to teach today, get out of here. Right? No, you hold your breath, right? And you hold your breath. Can you hold your breath forever?
No.
No, eventually you're forced to breathe, not because you run out of oxygen, but because you build up too much CO2. It is actually the carbon dioxide levels that actually cause you to breathe and not the oxygen. Yeah, exactly. It's not the lack of oxygen. It's actually the buildup of CO2. In fact, that's what I'm telling you. You'll determine whether that's true or not by doing that breathing control of respiration lab that we talked about. So again, doing that lab will help you to see if that what I say is actually true or not.
But what if you let your air out and hold, not letting you to take air in? What it's about not taking oxygen now.
Well, again, it does limit the amount of oxygen that you have. However, when you expel the air, you have definitely less oxygen in your lungs, but you also have less gas in your lungs for less gas exchange to take place as well. So once again, it is still the buildup of CO2, and we will see that. We will talk about that.
Okay.
Okay. Lastly, obviously, our respiratory system helps us to produce our sound. The movement of air across your vocal folds produce a vibrating sound that we use to produce our speech. We form that speech, yes, with our teeth, yes, with our lips, yes, with our tongue, but also with the resonance of our respiratory system and our nasal cavity as well, right? You can call a friend up on the phone and talk to them and they just say hello, and just by hearing their voice without seeing them or hearing them sniffle or anything else, you can tell they have a cold, because when they're congested, that congestion in the nasal cavity changes the quality of the sound of their voice. So both the physical ability to produce that speech and also to modify that speech is an important function of the respiratory system. A non-respiratory function, but an important function nonetheless. All right, questions on that? Excellent. Like most things in this class that we talk about, we can identify them in one of two ways. We can identify them anatomically, or we can identify them physiologically or functionally. And the same is true for the respiratory system. However, like many things, like when we talk about the joints, anatomical definitions, functional definitions are equally important. I would argue that the anatomical distinction is relatively stupid. And I think the functional is much more meaningful. When we talk about the respiratory system from an anatomical standpoint, as you can see here on the illustration, basically what we do is draw this artificial line through the respiratory system at the level of the glottis, at the level of the opening to the trachea. Right. I guess from an anatomical standpoint, this is, again, although this should be air only space, it's basically the head and up and then everything else down. However, is the function, for instance, of the trachea really any difference from the function of the pharynx or the function of the nasal cavity? No. So again, this is merely an anatomical distinction. The upper respiratory is basically the structures that are in the head, and the lower respiratory is basically from your Adam's apple down. So there it is. That's our anatomical. Again, other than being a distinction, it's not very meaningful. What is much more meaningful is the functional definitions, the functional distinctions, the structures that do different functions, I think is a much more significant way of dividing this material. And there are basically two main functions of the respiratory organs. The respiratory organs basically do one of two things. They either move and process the air, and actually let's write this. Move and process the air. Right, so these are things that are cleaning the air, moistening the air, watering the air, heating the air, all of those things as it moves to the respiratory zone, which is where our gas exchange takes place. So our respiratory zone is the functional region where gas exchange takes place, and the conducting zone is basically everything that brings air to and from the respiratory zone and processes it. And again, you may not have thought of this processing, but you know what happens, because if you're standing outside on a cold day and you breathe out, what happens? You can see your breath. And the reason you can see your breath is because you've warmed and moistened that air. So as it is expelled from your respiratory system, it hits that cold surrounding, it condenses, the water moisture condenses, and you see the breath. So again, you may not have thought of all the processing that takes place, but you're very much aware of it. All right, questions on that? Excellent. So again, more pretty pictures, even more pretty pictures. So let's work our way through the same way we worked our way through the anatomy of the digestive system by working through the path that food would take. Let's take the path that air would take through our respiratory system, starting first in the nose. The nose is really the only external respiratory structure. Air and fingers occasionally enter through those two holes, the openings known as the external nares. Entering into that entry point, basically formed by the rubbery connective tissues of your fibrous connective tissue and skin, that area known as the vestibule. So like I said, if you're picking your nose, not that I'm encouraging you, but if you ever see a 2-year-old pick their nose, their finger goes into the vestibule. That would be that region. The job of this is to obviously collect air, but we do need some protection in here as well. So what type of tissue do you think lines the entrance to our external nares?
Non-keratinized.
Yeah, non-keratinized stratified squamous epithelial tissue. And of course, what is going to be inside of it? What is the respiratory membrane of the nasal cavity?
Stratified ciliated columnar.
There you go. Ciliated. Pseudo. Oops. There you go. Yeah. Stratified. Fingers won't work today. Ciliated pseudostratified columnar epithelial tissue. Excellent. There we go. Perfect, perfect. Now, again, obviously we have that non-keratinized stratified squamous at the opening, a different one on the inside, but we also need that entrance protected by hair-like structures that help to stop large dust and debris and insects from entering into those. And of course, while we commonly refer to those as nose hairs, the appropriate anatomical term for those are the vibrissae. Vibrissae AE is the plural, just A, vibrissa is the singular. So those are those nose hairs that stick out. And in fact, I think if I remember correctly, if we go back to, there we go, excellent. Notice here in our actual cadaver, we can actually see, while we don't see the external nares, We can see the enlargement that is the vestibule. So this enlargement that sticks out laterally is the vestibule, and lo and behold, inside the vestibule, you can see those vibrissae. So you can actually see the vibrissae located on inside of the vestibule. So here's what I would say. It is not necessarily a bad thing to trim the vibrissae. There are two concerns that you have. The first is, obviously, if you're using scissors or something like that, you got to be careful about damaging the tissues. If you're using one of those electric nose hair trimmers, you could be irritating the inner lining of the nasal cavity that way. Remember, as we talked about with shaving, if you're irritating those, that can cause epidermal growth factor to be produced, which could increase the rate of growth of those hairs as a result. And again, you're getting rid of one of the layers of protection, potentially allowing larger dust and debris to be able to get access to your nasal cavity that way. So yeah, increase the likelihood of getting a bug up your nose, I guess. But again, it doesn't cause, I think, major concerns that way. And we just talked about the ciliated pseudostratified columnar epithelial tissue. Our nasal cavity is divided up into two parts. Unless you have done massive amounts of cocaine, you cannot put a handkerchief up one nostril and pull it out the other. Well, I guess if you have a piercing, you might be able to do that too. So cocaine and piercing are the only ways that you should be able to do that. But normally there is a structure that divides the nasal cavity in half. That, of course, is a skull feature of the skull. And what skull feature divides the nasal cavity into half? Nasal septum. Excellent. What are the bones and bone features that comprise that nasal septum? Excellent. Perpendicular plate of what?
Sphenoid bone.
There you go. Perpendicular plate of the ethmoid and the vomer. Excellent. There is also some cartilage in there, a little bit of fibrous connective tissue, but the bony components are indeed the perpendicular plate of the ethmoid and the vomer. One of the interesting things about the nasal cavity, you may notice at any one time, if you try to breathe between both sides of your nasal cavity, that air may not move as freely through both of them. Again, the reason for this, as we've talked about, is it is important to process the air in your lungs before it gets to your lungs for gas exchange. And one side of the nasal cavity may not be warm enough or may not be moist enough. And as a result of that, there is actually erectile tissue in your nasal cavity, similar, not identical, but similar to erectile tissue, like in the clitoris, like in the penis, like in the labia. And so what can happen is we can actually vasodilate those passageways to constrict them and resist air going through those and encouraging air to go through the more optimal side. And they can actually fluctuate in the dilation of these tissues to go back and forth. Like we talked about in the autonomic nervous system, these erectile tissues are controlled by or stimulated by the parasympathetic and inhibited by this sympathetic. So one of the things, like for instance, when you exercise, it typically becomes easier to breathe through your nose. When you take a cough medicine that suppresses the parasympathetic nervous system, that can help to vasoconstrict the nasal cavity, decreasing your mucus production and opening up your airways more. So that's what's happening with those in that. So we have that. And then, God, must have been like 10, 15 years ago now. It was a while ago. I never actually heard what happened to it. There was actually a group that was actually looking at different cognitive abilities in individuals when they were breathing through one side of their nasal cavity versus the other. And they claimed at the time, although like I said, it was like 15 years ago and I haven't followed up on anything since, that some certain people were better at, like for instance, solving math problems when they were breathing through one side of their nasal cavity through the other. So it was this really weird study that I read about, and I don't know whatever happened of it. Obviously, it couldn't have been that big of a deal. Otherwise, we'd all have plugs that we were using when we were taking exams or something like that. Well, so great question. A deviated septum is exactly that, where you have that structure that is supposed to be consistent on the midline. It can be curved, or it can be bent, or you can have some type of anatomical variation to those bones. And as a result of that, it is something that can affect your ability to be able to breathe freely through both sides of your nasal cavity. It can be it can be a problem for most people, but can be a serious issue for endurance athletes or hikers or things along those lines. And it is actually possible through reconstructive surgery to actually have a deviated septum either shaved off or reformed or broken and realigned to help with those. So in extreme cases, yeah, deviated septum can have major effects on breathing. It can affect respiration at night when sleeping. Certain forms of sleep apnea can be caused by severely deviated septums and things along those lines. So it can be something that can affect and influences someone's life by having that deviated septum. But there are ways to mechanically resolve that. Yeah, so again, it's not uncommon that way. Alrighty, excellent. Oops. And again, we already talked about our mouth, so I won't beat a dead horse with that one. Again, the nose is associated with the paranasal sinuses. So as we know, if you hopefully remember from 430, there are, so actually I need that over there so I can grab this. There are 4 bones of the skull, 4 foot bones, 4 skull bones that have sinuses that are associated with the nasal cavity. Anybody remember what those 4 bones might be? Excellent. Frontal was one of them. What else? Ethmoid. What else? Maxillary. And sphenoid. Excellent. These 4 bones all have, and I think I have a pretty picture that shows this. There we go. So let me grab my writing and move it up here out of the way. Here in a frontal and a sagittal view, we can see those sinuses and their locations. These paranasal sinuses help us to produce turbulence. Right? You could be standing by a side of the river, and that river could be flowing very, very rapidly, but the surface of it can still be relatively calm, so that you could put your boat on it and very quickly get down the river. But that's not nearly as much fun as instead when there are rocks and logs and things like that in the river. Because when there are those uneven structures in the river, that produces the white water, that produces the turbulence. And again, as we talked about, we don't want the air just going straight down into our lungs. We want to swirl it around. We want it more in contact with our mucous membrane so that we can clean it, we can warm it, we can filter it, we can moisten it, we can do all those things to the air to clean it and process it before it reaches the lungs. And so having these paranasal sinuses allow us to produce that turbulence, swirling that air around. And of course, also swirling the air around helps it come in contact with the superior surface where our sensory structure is located. And anybody remember what the sensory structure that allows us to smell is? Olfactory nerve, absolutely. If you remember from 430, there is an enlarged structure called the olfactory bulb and olfactory tract that come out of our limbic system of our brain. And that bulb of the limbic system sits on what bone and bone feature again? Not the coela turcica. What bone feature does the olfactory bulb sit on? Well, getting closer. So it is indeed the ethmoid bone. However, remember the bulb sits on the cribriform plate. But you are correct in that the olfactory nerves pass through small holes in that cribriform plate. And those small holes in the cribriform plate are the olfactory foramina. Because of course, foramina is the, foramina. Oops, no, hold on. Foramina. There we go. Is the plural of foramen. And it passes through into the nasal cavity, providing us with our sense of smell. Excellent. Notice, I love this picture from your textbook. This is a posterior view of a coronal section. We can see the roof of the mouth, that hard palate of the oral cavity, as we talked about in our digestive system, is made up of the two maxillary bones and the two palatine bones. Here we can see some of the nasal paranasal sinuses. Here we see the maxillary paranasal sinus. Here we see the frontal paranasal sinus. So we can see that. We can also see some of the air cells that make up the ethmoid sinuses. But notice we can also see some other structures that allow us to produce turbulence as well. These big bumps located in the nasal cavity. Oh, and beautifully we can see the nasal septum as well, perpendicular plate of the ethmoid and the vomer. This bump up here, anybody remember what this bump up here is called? If you're not certain, you can always cheat by reading it off of the, there you go, superior nasal concha. Excellent. And remind me again, is the superior nasal concha a bone or a bone feature? Is it its own individual bone or is it a feature of another bone? It's a feature of another bone. Which bone? Ethmoid. Excellent. What's this bump called? Middle nasal concha. Excellent. Bone or bone feature? Bone feature. What bone?
Ethmoid.
Ethmoid. Excellent. And what's this third bump called?
Inferior.
Inferior nasal concha. Is it a bone or a bone feature? Bone. It is its own bone, one of the facial bones. Excellent. So again, these produce turbulence. And notice they also produce these small passageways between them. So the passageway between them is what is known as a meatus. So the same way we have a superior, middle, and inferior nasal concha, we have a superior, middle, and inferior meatus. We see it really nicely here in this sagittal view. Here we see those exact same things with a, pardon me, in a coronal view. Here we see it in the sagittal view, where again, there is that middle superior, pardon me, the superior and middle nasal conchas that are part of the ethmoid bone. Here is the inferior nasal concha. And again, those meatuses are the spaces between them. The sphenoid sinus would be located right here. Notice we see the frontal sinus nicely there. So air enters into the nasal cavity through the external nares, gets swirled around by those paranasal sinuses, by the concha, producing all that turbulence, swirling it around. Then it finally exits the nasal cavity into the nasal pharynx out of an opening known as the internal nares. And I believe, if I don't have it here, I don't, so let's cheat. Hold on. I need to do that. And then I need to do this. Oh, I know which picture I like better. I like that better. All righty, I'm stealing a picture from my 430 class where we were looking at the bones. Notice, can you guys see the bones now? Notice as we look at this inferior view of the skull, here we see our hard palate made up of the two maxillary bones, made up of the two palatine bones. And then notice here, the palatine bone, which we know is this kind of L-shaped bone up here. And then here we have a little bit of the vomer, and here we have the sphenoid. Notice this opening in the bone. We see the same thing here too. This opening in the bone right here, this is that internal nares. So this internal nares is the bony restriction that limits the movement of air out of the nasal cavity into basically what here would be the nasopharynx. All right, so we saw you in the lecture as just this line that's drawn between the end of the hard palate and basically the wall here, which would be the sphenoid, but we can actually see that it is these restrictive bony openings that are known as the internal nares, the opening into our nasopharynx. And luckily the pharynx is an organ we know. All right, questions on that?
So where is the external nares?
External nares is where your finger goes.
Oh, yes. Okay.
Yep. All right, and I don't mean your finger. I mean Eric's finger. All right, excellent. Questions on that? All right, excellent. So again, as we talked about lining our nasal cavity, we have that ciliated pseudostratified columnar epithelial tissue that is going to move the mucosa to the nasal pharynx, where it can either be swallowed and spit out. So as we talked about, we know we have that ciliated pseudostratified columnar epithelial tissue. And again, we know that is going to be in the inside the nasal cavity and in the nasal pharynx. But notice air also has to pass through the oropharynx and the laryngopharynx. So what epithelial tissue would we have lining those? Yeah, maybe it helps if we go back to the picture. There we go, excellent. So, excellent, all right, perfect. So let's cheat. Our non-keratinized stratified squamous epithelium. Excellent. Perfect. Of course, then when we get back into the larynx and into the trachea, we're going to have that ciliated pseudostratified columnar epithelial tissue again. But when we get all the way down to the alveoli, what type of epithelial tissue do you think we're going to want lining that? This is where gas exchange is going to be taking place. So we want to make that alveoli as small of a structure as possible. Absolutely. So we're going to have a simple squamous epithelium. And so do you think that just like halfway down our respiratory system, in our lungs, it suddenly goes from a ciliated pseudostratified columnar instantly to a simple squamous? No, it's going to transition. So along the way, it's going to go, so let's see, ciliated pseudostratified columnar to non-keratinized, back again to a ciliated pseudostratified columnar epithelium. And then from there, it transitions to a simple columnar. And from there, it transitions to a simple cuboidal. Until finally, it gets down to that simple squamous epithelial tissue in our alveoli for the gas exchange to take place. So it starts all big and protective, and then it gets smaller and smaller and smaller in this simple transition as it works its way down the conducting zones. Of course, here we see that ciliated pseudostratified columnar epithelial tissue with those big mucus-producing glands. As we talked about, it helps to both move the mucus and the dust and debris to our nasal cavity to be swallowed and spit out. And from our larynx and trachea, it moves it up, actually forming what we call a mucus elevator to bring that mucus up and to be able to release it. Again, as we talked about, these cilia are delicate. So for instance, if you happen to be a chronic smoker, the warmth and the toxins and the irritations of that smoke can damage these cilia. So they become non-functional. So how can you tell when you've been around someone who's a chronic smoker? Well, they build up this dust and debris inside of their airways, and how do they get rid of it?
Yeah.
Coughing. That nice that smoker's hack that you get as a result of that. That's how they release that dust and debris. Actually, let me answer your question about that in just 1 second. But one of the concerns for someone who is a chronic smoker is they get a cold, or they get a little bit of the sniffles, or they get their allergies, and they take a cough suppressant. If you take a cough suppressant, now you're suppressing that cough reflex, and your poor little cilia can't work. So what happens to all that mucus, all that dust, and all that debris? Drains down into your lungs, dramatically increasing likelihood of getting some type of respiratory infection or pneumonia, or even ultimately leading to to cancer and stuff like that. All right, Ashley, what's your random sneezing question?
Why is it that some people can look at the sun and it helps them sneeze, and some people look at it to make it go away?
Because my guess is that neither of it has anything to do with the sun. It has more to do probably with the change in the position of the head.
Right, yeah.
Yeah, my sister has always been one of those that claims that staring at a light stops her from sneezing. But she also believes that saying the word watermelon stops her from sneezing as well. So I guess if anything, it's those things are moving the head or moving whatever. And so the hope is that whatever that irritation is shifts and doesn't irritate you as much anymore.
Okay, got it.
Yep. Yep. So here's what I would say, that the primary damaging component to the cilia is the warm smoke. Right? When you're burning something, that burnt aspect of it is what irritates the cilia. What I don't know, not that I'm encouraging everybody to run out and start vaping, but I don't know if vaping, because it is more of a cool misting type of a thing, if it irritates the cilia in the same way. So it's more the burning of the smoke. So it wouldn't matter what you were smoking, whether you were smoking tobacco, cloves, marijuana, anything else that you were smoking. I think it would have it has similar effects on the cilia. So it is more the temperature and the burning of it that is what causes the irritation to the cilia. But I'm sure there have been studies that have looked at the effects of vaping on the cilia, but I'm not familiar with them. When we get to the anatomy, we'll talk about where there's the transition in the epithelial tissue. So we'll get there. It's a great question. I don't have an answer for that. My guess is that the reflex of closing our eyes when we sneeze is probably more protective so that we're not getting any of those propellants that are being released out. I think they have done studies that show that if someone has their eyes forcefully held open, they are capable of sneezing. So it's not like you can't sneeze with your eyes open. It's just, it's incredibly hard to do because it's a very powerful reflex. And my guess is that it's a protective reflex. Related to the contraction of the muscles for that causes that. Okay, well, so there you go. There you go. Exactly. And like I said, I know they've done studies where they forced people's eyes open and had them sneeze. And it is possible to sneeze with your eyes open. But so my guess is that it's, like I said, it's probably more of a protective reflex. All right. Excellent. Fun questions. I like this. This is good. All righty. But this is a good point for our first break. Well, actually, let's do this real quickly first. So just to finish off the tissues. So as we've seen, our respiratory passageways can be lined with all sorts of different epithelial tissues. However, regardless of what epithelial tissue is on top, what type of connective tissue is always underneath?
Alveolar.
Alveolar, excellent. And what would we call this areolar connective tissue based on its location?
You mean appropriate?
There you go. This is, again, a mucous membrane open to the outside world. So based on location, we would call it a lamina propria. All right, because after all, the membrane is a mucous membrane. Our respiratory passage is open to the outside world. Excellent. In our upper respiratory tract, again, we have lots of mucus glands to capture that dust and debris and to moisten the air. Also in our upper respiratory tract, especially our nasal cavity, we have a lot of very superficial blood vessels. What is the purpose of those superficial blood vessels? Why do we want a lot of blood vessels near the surface of our mucous membrane in our nasal cavity? To warm the air. As we know, blood is 2 to 3 degrees warmer than our core body temperature, so it's able to radiate out lots of heat to moisten that air. Molecules at a higher temperature move more quickly. So if we can warm the air, diffusion of those gases will happen more rapidly. Of course, what's the downside of having all those close line superficial blood vessels in the nasal cavity? Right, Marcia hits you in the nose with a football, right? It gets all big and swollen and bleeds as a result of that. Yes, when they talk about having an upper or lower respiratory infection, typically it's one in the head or one in the lungs or the bronchi. That is correct. So again, I'm not saying that it isn't necessarily meaningful to have that anatomical definition, but you know, for those kind of functions, but I think the functional makes much more sense. You don't want to cool the air. We don't want to cool the air. We want the air warm because, again, we're not using any ATP to get that oxygen or get those gases to diffuse across the membrane. So we want it as warm as possible so those gases are moving as quickly as possible. So yeah, so we don't want to cool the air. Well, if the air coming into your nose was above your own body temperature, then I guess technically the blood vessels would draw away some of that heat because the heat's going to go down. But yeah, but it's also very moist in that steam room as well, so that's a good thing. Great question. So Bell's palsy is more of a condition with an infection of the mastoid sinus. So it wouldn't necessarily be cold air coming in through the respiratory system because the respiratory system doesn't connect to that mastoid sinus. Although there have been issues of, I know of a couple instances that have been recorded of driving with the window down where the air is blowing by and that irritation of the air blowing by is caused Bell's palsy. Bell's palsy, of course, is constriction of that facial nerve through the stylomastoid foramen of your temporal bone, but that doesn't connect to the nasal cavity. Certainly, the colder it is outside, the harder it is for you to warm the air as it comes down, and so that could be a sensation that you feel from that. Yep. Excellent. And then we already talked about the olfactory receptors, how they're the olfactory nerves located up in that epithelium underneath your cribriform plate. One of the interesting things about your olfactory nerves is your olfactory nerves only last for about 7 days, 7 to 10 days. They're constantly being replaced. When this was first discovered, respiratory neuroanatomists were all aflutter because they're, here in our body, we have nerves that regenerate and are constantly being replaced. So why can't other nerves regenerate in the same way? And so a tremendous amount of time, thought, effort, money, research went into studying these olfactory nerves as what's so special about them that they can regenerate and other ones can't. Because if we could unlock the key to that, then maybe we could get other nerves to regenerate as well. But the research never really went anywhere, and we started having much more success with things like stem cells, embryonic stem cells and things like that. So the research shifted much more in that direction. But at the time, those olfactory receptors, when it was first discovered, was a very, very interesting field of study.
I have a question.
Yes.
How it works when I'm cooling down something, like when I'm cooling down the scoop of the hot soup with my air? How can I do that?
Okay, well, so the things that I would say is if you have a bowl of hot soup or if you have a cup of hot coffee, those things are much closer to boiling in temperature. So those things are, closer to 200 degrees Fahrenheit, as opposed to your core body temperature, which is, 98.6 degrees Fahrenheit. So when you blow air out of your lungs, even if it's a couple degrees higher than that, like 100 degrees, it is still going to be much cooler than the fluid that you're blowing over the surface of. And the other thing that you're doing is you're producing a current. So it isn't even so much just the air that you're producing, because if you think about it, if you just wave your hand over it, that would cool it as well, because you're producing a current. And as that air moves across the surface of that, it steals heat away from that radiating heat of that substance. So it's, you're just blowing is an easy way to swirl it takes less energy to blow than it does to sit light and do this over your coffee. Although this probably would be more efficient.
But there's a difference between like temperature of the air. If I close my lips and let it like go just a little, it's cooler than if I breathe like with the whole mouth,
Yeah, because you're swirling more air around as well. Absolutely.
Okay, thank you.
All right, excellent. You guys got some good questions today. I like this. Perfect. All right, this is a good natural stopping point. We need to move on to the pharynx and talk about what's happening there, but I think this is a good stopping point. So it's 1:18 now, so let's go ahead and take our first break, come back in 15 minutes, 1:19 now. So that makes it, let's call it an even 1:25. So 1:25, we'll restart, and I will start the recording at that point. All right. Questions on any of that? All right, see you in 15 minutes. All righty. Sorry about that. Let's go ahead and get started. So we've been working our way through the passageway, starting first in the nasal cavity. And as we mentioned, it enters into the pharynx, which is that muscular air passage. And again, we talked about this when we did the digestive system. We know that the nasopharynx is for a passageway for air only. But as we also talked about, the oropharynx and laryngopharynx are passageways for food, drink, and air. And so as we talked about, there are differences in their epithelial tissues. The nasopharynx, like the nasal cavity, is primarily lined with a ciliated pseudostratified columnar epithelial tissue, whereas the oropharynx and laryngopharynx are lined primarily with that non-keratinized stratified squamous epithelial tissue. All right, so we did all of that. And again, that's some new information. We got that in the digestive system. And then we have it here. The only thing that, again, I would like to emphasize, and again, these are things we talked about both in the digestive system, but we also talked about it in the lymphatic system as well. The location of our tonsils in relation to this. Remember, we have that pharyngeal tonsil located up here at the superior boundary of the nasal cavity. Remember also we have that opening to the auditory tube that is located in the nasopharynx. We talked about the arches and everything then in the oral cavity and the oral pharynx and things along those lines as well. So again, this is anatomy we should be pretty comfortable and familiar with as we talk about those things because we've seen them before. The other thing that we talked about is we talked about the function of the larynx in its role of that deglutition, that swallowing reflex. We learned about how we use those muscles to elevate it, that closes the epiglottis and allows the air to pass through. And we talked a lot about the epiglottis as one of those important structures in that deglutition reflex. However, what we didn't talk about is more of the rest of the anatomy of that larynx as well. As we talked about, it is lined with a ciliated pseudostratified, columnar epithelial tissue. And this doesn't have to be quite that big anymore. But there are two folds in the lateral walls of the larynx. These two folds are what are commonly referred to as the true and false vocal cords, but are more appropriately referred to as both the vocal fold and the vestibular fold. So let's take a look at those. So, oh, I lied. I thought I have a good picture of those. I do it in different. Okay, so we'll talk about those in a second. So we have not the vocal cord, but a vocal fold, which is the inferior fold. And above that, we have what is known as the vestibular fold. I do have some pictures that will show this, but I want to talk about the anatomy of the larynx first. So within the mucous membrane of the larynx, we have these two folds in that pseudostratified ciliated columnar epithelial tissue, the vocal fold, which, not surprisingly, is what we use to produce our speech, and the vestibular fold, which protects it primarily, as well as protecting our lungs. But we'll talk about those anatomy in a minute. Let's talk first about the larynx. If you were to rip the larynx out of your throat, what you would see is that it is a cartilaginous structure. Remember, most of the structures that we talked about in the digestive pathway were all muscular tubes, so that when they're not in function, they can collapse, like the esophagus can collapse when it's not being used. However, we are hopefully constantly bringing air in and out. So it's not surprising that we want to have a rigid structure that is going to help to maintain and hold its shape. Notice also associated with the larynx, but not actually a part of the larynx, is that hyoid bone. And someone remind me again, what's the special thing about the hyoid bone?
Base of the tongue.
It is the movable base of the tongue. Ashley, what do you mean by it floats? I like that description and it's fairly accurate, but what do you mean? Can you be more specific? There you go. Excellent. Right. All the other bones of our skeletal system form at least one joint, at least one articulation. The one exception is the hyoid bone. The hyoid bone is only held in place by connective tissues, cartilage, membranes, ligaments, tendons, things along those lines. But it does not actually articulate with any other bone. Excellent. Perfect. So this cartilaginous structure is comprised of 8 rigid hyaline cartilage features that again give it shape, give it structure. And then as we also talked about, we have that 9th cartilage, the epiglottis, which remember is made up of elastic cartilage, allowing it to flop over the top. Here we get a better view of this. And remember, as we talked about and mentioned before, the thyroid cartilage is the largest of all the cartilaginous pieces. it has a large bump that sticks out prominently anteriorly, what we commonly refer to as the Adam's apple, but the appropriate anatomical term for it is the laryngeal prominence. So do not use Adam's apple on the exam. You will not even get partial credit. However, why is it called the Adam's apple? Anybody know? Excellent, because it's bigger in men. The reason for this is actually those androgens. Typically, when a boy hits puberty and they start producing those androgens like testosterone, it causes a thickness of the bones, it causes a thickness of the muscle, and it causes the cartilage here to grow as well. So typically that Adam's apple becomes a prominent feature, more prominent thanks to those androgens, and is often seen as a desirable characteristic. It's an indication of high levels of androgens, meaning you are more manly, right? Notice one thing, while it is the largest hyaline cartilage piece of the larynx, notice as you see from this posterior view, it doesn't go all the way around. It is a shield-like, C-like structure where it has that prominent front with that laryngeal prominence on the front and then those lateral aspects, but it is actually open on the posterior side. In fact, as we'll see, there's only one piece of hyaline cartilage that is completely circular for the larynx. Well, I think it does come from the biblical reference of Adam from, eating the apple in that, but I think, again, the point of it is that it is more prevalent in males than in females. And so since Adam was, biblically the first male, that's why it's called the Adam's apple. So, but it's more prominent in males. And so I think that that's where the reference comes from. You are correct. So it does have to do with that. True. Well, and then also apples didn't exist back then. It was probably more likely some other type of fruit as well. But again, this isn't that place for that type of philosophical debate. Excellent. As I mentioned, there is only one piece of cartilage in the larynx that is completely circular, completely enclosed, and that is the cricoid cartilage. The cricoid cartilage forms the circular base of the larynx and is what actually connects it to the trachea. The trachea is a muscular tube as well, and I will show you a quick cheat. Notice as we look at it from the front of our trachea, it has these cartilaginous rings, but notice as you look at the posterior view or the side view, you'll notice that the rings of cartilage in the trachea are also C-shaped. They do not go all the way around and connect. So really the only piece of cartilage in this entire airway that is completely circular is the cricoid cartilage. Notice also the cricoid cartilage is smaller in width on the anterior side and larger on the posterior side. That is in part because on the posterior side, it forms the base for these two movable pieces of cartilage. These two movable pyramid-shaped pieces of cartilage are what are known as the arytenoid cartilages. These are connected to the vocal cords. So what happens is when these arytenoid cartilages rotate, they are able to open our airway, open our vocal folds, open our airway, allowing us to express that air, allowing us to vibrate our vocal folds, or like we talked about in digestion, we can also use them to hold the glottis closed to let us use that Valsalva maneuver to help in defecation. So these two cartilages, along with the tiny little corn kernel cartilages on top, and that's of course how I remember them, these tiny little nubs of cartilage on top of the arytenoid cartilages are called the corniculate cartilages. Those four cartilages, the two corniculate on top of the two arytenoid, are what help to open and close the airway, open and close our vocal fold, open and close the opening we call the glottis. And we will take a look at that in just a minute. It's a great question. My guess is that it have something to do with flexibility. Also, remember, posterior to this is the esophagus. So if you think about it, I know everybody here takes a nice reasonable sized bite of food and chews it the 30 times you're supposed to before swallowing it down. But some people will take half a cheeseburger in one bite, chew twice, and swallow it down. And that, if you think about it, that flexibility might allow for movement of larger boluses into the esophagus in that way. So again, if you had a posterior, really large posterior rigid structure, it might restrict the passageway of food. And so it may play a role in that. Again, remember, why is always one of those questions that it's hard for us to answer. You know, the engineer who designed all of this didn't leave us her footnotes, but we can kind of infer sometimes based on the function of things, why it might be beneficial that way. Now, notice my math. How many pieces of hyaline cartilage did I say were present here in our larynx? 8. How many have we counted? Well, 6. Thyroid cartilage, cricoid cartilage, 2 arytenoids, 2 corniculates. So that's 6. So we're missing 2. And the reason we don't see those 2 of them here is because they're not present in this image. Because what this image is lacking is our mucous membrane. Obviously, this is the cartilage, but the other thing we need to realize is this epiglottis needs to fold over the top, closing that airway. And it is part of a membrane that comes and wraps around all of this, that basically goes from, well, actually here, let's do a better job of this. Kind of goes to the arytenoid cartilage around this way. So there is a special fold in the mucous membrane that goes between the arytenoid cartilages and the epiglottis. Any idea what that particular membrane might be? That fold in the membrane might be? If only I had a list of the anatomical terms I was responsible for in front of me, I might be able to figure out what that fold in the mucous membrane between the arytenoid cartilage and the epiglottis might be. There we go, the airy epiglottic folds. Excellent. We don't see it in this picture, but notice if we change our view, here we can see that membrane, that fold in the membrane. So notice here, we have our mucous membrane wrapping around the epiglottis and coming down and lining the outer surface of the arytenoid cartilage. And then there's that little corniculate cartilage. And notice in that airy epiglottic fold, we actually have an additional piece of cartilage that is solely embedded within that membrane. It helps to give more structure and support. It helps in the folding of that membrane when the epiglottis comes down, but it is not connected to any of the other cartilages. So that way, if we had a picture like this, it would have to just be basically hovering in space, like right there. It would just be hovering in space right there because, or hovering in space right here, because there's nothing to hold it in place. However, if we look at some of the other views we've had for this, and I don't remember if I have it here. Hold on one second. I don't believe I have it here, but let me double check. And if not, I've got the perfect picture for it. There we go, perfect. So, remember when we were on that virtual anatomy lab at Cosumnes River College and we were looking at some of these systems, we were looking at the half head chart and we've seen these things here. And remember, we were looking at this posterior view. Remember, we were looking at this before because it showed us the thyroid gland and the parathyroid gland, and that was where we were emphasizing it before. We used this to talk about the epiglottis and the esophagus and the uvula when we were talking about the digestive system. But notice if you look closely at this picture, one of the things that you will notice, and I'll use my highlighter for this, and let's use a light color like yellow. Notice that here is our epiglottis. Here is our aryepiglottic fold that is coming down. And notice on the posterior part of the larynx, we have 1, 2, I'll make them yellow, 3, 4 bumps. The first 2 bumps, the ones that I made in yellow, would be those corniculate cartilages that are sitting on top of the arytenoid cartilages, where the 2 superior and lateral bumps are the cuneiform cartilages, those ones that are just embedded in the membrane. So if I clear all of this, notice you can clearly see those 4 bumps. The 2 corniculate, which are medial and inferior, the 2 cuneiform, which are superior and lateral. So many of the models, many of the charts like this on the posterior side of the larynx, you will see those 2 bumps if they have the aryepiglottic fold. Only if they have the aryepiglottic fold can you see those 2 cuneiform cartilages embedded within it. All right. Questions on that? All right, excellent. As we talked about, the job of the larynx is not just to close up and protect the airways when we swallow, but also, as I said, to use those arytenoid and corniculate cartilages to rotate open the vocal folds. And that is what's going to allow us to be able to let out air and to also produce our speech. And here we have the pretty picture that I was looking for before. So notice a couple of key features. This is a superior view. We are looking down into the larynx from a superior view. So again, up here we have the epiglottis. Here we have the aryepiglottic fold. And notice once again, 1, 2, 3, 4 bumps in them. The 2 medial ones are the corniculate cartilages, and the 2 lateral ones are the cuneiform cartilages. But the other thing that I want you to notice when you're looking at this is that we have 2 overlapping folds in the mucous membrane. This fold that we see here is the superior fold that we see all of it from this view. And this is the vestibular fold. This vestibular fold is also lined with a ciliated pseudostratified columnar epithelial tissue. Right, it is a softer tissue underneath it, and it is highly sensitive. If dust or debris come in contact with this vestibular fold, it triggers a violent coughing reflex, so that if any dust and debris get that close to our lungs, we can try to force that dust and debris out. So it's highly sensitive. If you've ever had water go down the wrong pipe, it often comes in contact with this vestibular fold and you cough it out explosively as a result of that. Notice it sits on top of a much more rigid and yet flexible membrane. And that rigid yet flexible bendy membrane that we have there is our vocal fold. And that vocal fold is what we vibrate to produce our speech, ultimately. So again, it's that nice, inflexible, doesn't vibrate the same way, superior and lateral, and that very elastic inferior medial one that we are able to vibrate. How we vibrate it determines our speech. The bigger the vibrations we cause, what happens? If you increase the size or the amplitude of your vibrations, not necessarily a deeper voice, what do you get? So again, let's be careful. So if we change this, so again, we're producing sound waves. So if we increase the amplitude of the waves, and by amplitude, I mean the size of the waves, if we vibrate them bigger, we get a louder, excellent. Increasing the amplitude or the size of the waves determines the volume, right? However, you've got the right idea. If we change the frequency of the waves, then that affects the pitch. And if we make the frequency a higher frequency, what happens to the tone of the voice? Higher frequency gives us what? When we vibrate it faster, we get, well, what do you mean by more tone? Does the tone increase, get higher, or does it get lower? Higher frequency, higher pitch, exactly. Whereas if we use a lower frequency, we get a lower pitch. Right? How many of you play the guitar? It's the exact same thing that when you tighten or loosen the string of the guitar. The more you tighten the string, the higher the tone, the higher the pitch. The more you loosen it, the deeper the tone, the deeper the pitch. Absolutely. So we can change the tone to make it a higher tone or a lower tone by changing the vibration. And we all take this for granted, how challenging this is to be able to control it. It is something we voluntarily control. It's subconscious. You're not actually thinking of which actual muscles you are contracting, but you're absolutely voluntarily contracting those muscles in just the right way to produce just the right amplitude and just the right frequency to produce the sound of the voice that you want. And of course, what's the easy way we know how this occurs or that that's the case? Well, let's go back to that boy in puberty again. One of the things that happens to that boy in puberty is not only does his cartilage thicken, but the vocal fold thickens as well. And when the vocal fold thickens, when you vibrate it, does it necessarily vibrate at the same speed or amplitude that it was doing before? No. And so does it take that boy a little bit of time to relearn how to properly control those muscles to produce appropriate speech? Yeah. So what happens to that growing young lad as he is entering puberty? His voice cracks. Absolutely. That voice cracking that occurs because of that change in the vocal fold, and they need to learn new control of those muscles. Now, one more quick important question. If I were to take a microphone and actually insert it into your throat, right down here where these vocal cords are located, and I asked you to say your name, aside from the fact that you have a microphone shoved down your mouth, and when you said your name, would I hear your name? No, exactly. What these vocal folds actually do is produce a buzzing sound. If you took a microphone and put it down here, all you would hear is this buzzing sound. That buzzing sound that is produced is what we call phonation. Then, as we talked about, we use our tongue, we use our teeth, we use our mouth, we use our nasal cavity to shape that buzzing sound into meaningful words. And that shaping of that buzzing sound is what we call articulation, right? If someone holds your tongue, you can speak, but they may not be able to understand you all that well. Because while you can produce the appropriate sound, the phonation, you can't shape it appropriately into meaningful words. Right? For a while there, again, I guess with COVID, it's no longer popular, but for a while there, those mouth guard things that they put in, that the dentist puts in your mouth when he's cleaning your teeth, right? People were wearing those and trying to say sentences. There was a whole game you could buy that had a couple of those things in it, where you'd put those things in your mouth and try to say, you know, Polly want a cracker or whatever it was. And with your mouth being held open, you wouldn't be able to produce that, articulate that, and people had to guess what you were trying to say. It's a great question. He was quite known for having prominent teeth. I think also the singing also has to do with the phonation, how well they can produce that sound there. Because I think if you think about it, there are many musicians who have had, like, for instance, what's his face from Aerosmith. He strained his vocal cords and had to rest them. Julie Andrews, thank you. Julie Andrews famously had polyps on her vocal folds that she has had to have removed, and now refuses to sing. Allegedly has lost her ability to sing and doesn't sing anymore as a result of that, because polyps have formed on that. So obviously the ability to shape it makes an effect, but obviously how you're producing that sound has a big effect on that as well. So I never thought of that in terms of Freddie Mercury. That's awesome. Sure. Gene Simmons had his, again, allegedly, I believe either tore accidentally or actually purposely had his lingual frenulum cut, which allows him to project his tongue. Although when I think of great singers, Gene Simmons isn't necessarily who comes to mind, but I get the idea. So, great question. So some people are able to talk or sing. Ventriloquists are great at doing this, but the trick with that is that they are producing a different types of sounds. If you actually look into or talk to or hear some of the interviews that people do with ventriloquists, when they try to say something, they don't actually say the word. They make a different series of sounds that when their mouth is closed, when their teeth are closed, right, they're able to shape it differently, and then it comes out sounding like whatever word they want to do. But there is a whole science to how they are articulating their phonation to trick us into thinking that those words are coming from somewhere else. Yep, there's a whole science to that. All right, excellent. So, any other questions on the larynx before we move down to the trachea? All right, here we see that anterior view of the trachea, what we commonly refer to as the air pipe or the windpipe. It is an air passageway that leads from the larynx to our bronchi. Again, it is lined with that ciliated pseudostratified columnar epithelial tissue. And in this case, the cilia move upward. And I think I've used that term before. It forms what we call that mucus elevator to help to take that mucus up and out of away from our lungs. It terminates at a branching point, and at the base of that branching point is a sensitive region known as the carina. Remember how we talked about those vestibular folds are heavily innervated, and when an irritant, dust or debris or water or something else comes in contact, it causes a violent coughing reflex. Well, the carina is the same way. The carina is basically like that last line of defense. If that water goes down the wrong pipe, if you inhale some kind of dust or smoke or something along those lines, if those irritants reach the carina, it produces, again, an incredibly violent coughing reflex in hopes of expelling whatever that irritant is and making sure that it doesn't reach the lungs. So we have this very, very sensitive mucous membrane at the base of the trachea in a region known as the carina, which again causes that explosive coughing reflex to try to limit the damage of substances getting into your lungs. That single trachea branches into two primary bronchi. And why do we have two primary bronchi? Because we have two lungs. There you go. By sensitive in this case, I mean that it has some very sensitive tactile receptors, absolutely. So that if something comes in contact, or chemical, both chemical and tactile receptors. So if some type of irritant or some type of chemical comes in contact with it, it causes us to have that explosive coughing reflex as a result to it. Because again, by the time the air is getting to the lungs, it should be relatively free of any type of irritants. It should just be that clean, warm, moist gas. All right, excellent. Notice also, clear that. If we look at the trachea in a cross section, we can see how I mentioned that those cartilaginous rings that we saw here are these cartilaginous rings that line our trachea, are indeed C-shaped. They do not go all the way around. They instead just go around about 3/4 of the way around and do not meet in the posterior. Posterior to it is where we have that esophagus. And as we mentioned, that esophagus is a muscular tube. Its job it is to be able to expand to accommodate the bolus. And so we don't necessarily want that bolus getting caught on a big ridge of cartilage. So it makes sense to have it open in the back to allow for more expansion for air to come through and food to go through without dislodging and affecting our ability to collect air. However, do we need to be able to modify the size of the trachea? Yeah, so having a ring of cartilage would not allow us to do that either. So by having it open, if you'll notice, there is a thin piece of smooth muscle that connects the two posterior parts of the cartilage ring together. Here we see that little bit of smooth muscle here as well, and it is known as the trachealis. So that trachealis muscle gives us some ability to be able to expand or constrict our trachea, opening and closing our airways. So again, the other advantage of not having an enclosed ring of cartilage is if you had an enclosed ring of cartilage, we wouldn't really be able to change the shape. By having it C-shape, we are able to constrict or relax that muscle, and it gives us some limited, but still some ability to change the shape of the airway. So there you go. We talked about all of that. All right. Questions on that? All right. As we mentioned, we have two primary bronchi because we have two lungs, and they go to the two lungs, and that is the key. The two primary bronchi go to the two lungs, but they do not enter the lungs. Instead, they branch first before they enter. Oops. And guess what the primary bronchi branch to form? What do you think primary bronchi branch to form?
Secondary.
Secondary bronchi, excellent. Before they enter. And do we just have a random number of secondary bronchi? What do you think? No.
Maybe.
No. How many secondary bronchi do we have? Anybody know? So we have two primary because we have two lungs. I like the 5 answer better. Let's go with 5 secondary. Secondary. Anyone know why we have 5 secondary bronchi? Well, we have 3 lobes in one lung, and then how many on the other lung? 2. There you go. So we have a total of 5 lobes to the 2 lungs. And then it is these secondary bronchi that are going to enter into the 5 distinct lobes. So the secondary bronchi enter, 1 per lobe. There you go. Again, why is always one of those tricky things, but definitely is there a different morphology between the left and the right lung because of the location of the heart? Absolutely, Alex. So I think it's reasonable to say that is because of the location of the heart. So let's actually look at this. Let's actually look at the lungs. There we go. Well, actually, we'll talk about that first. Before we do that, one last thing to point out about the primary bronchi. not only are they going to different number of lobes, but there is a morphological difference here. The right bronchi, and again, partially because of the location of the heart, but the right primary bronchi is typically shorter, wider, and more vertical, whereas the left one is longer, more narrow, and more obtuse in its angle. From a clinical standpoint, why might that be significant? Why might the anatomy of the two bronchi make a difference? Yeah, absolutely. If you happen to accidentally, because you're eating a big bowl of blueberries, and you accidentally inhale one because you try to talk while you're eating a blueberry, and it enters into your trachea, is it equally likely to go into the right primary bronchi and the left primary bronchi? No, it's much more likely to go into the right because of the shape, the size, and the orientation. Dust and debris, materials, blueberries, are more likely to go into the right. So from a clinical standpoint, if something needs to be removed, typically they look for it on the right side first. All right. Excellent. All right. Let's talk lung anatomy. With our lung anatomy, we have, of course, two lungs. The lungs are cone-shaped organs. And what's a key to a cone shape? Our heart was cone-shaped. Anybody remember what the anatomical characteristics of it is? Excellent, absolutely. A cone-shaped organ like the lungs have a wide, round base, flat base, and then it comes up into a tailored peak-like structure. And as you guys mentioned, the tailored peak is the apex, and the broad flat part is the base. Remember, our heart had this cone-like structure as well, but with the heart, remember, it was rotated. The base was superior, and the apex not only was inferior, but remember, it also projected anteriorly and also slightly to the left as well, so projected out that way. But in this case, with the lungs, the apex is up, the base is down. Now notice the big broad bases of our lungs sit on top of an important organ. What is that nice curved bell-shaped organ that the lungs sit on top of?
Diaphragm.
Diaphragm. We can see it here sitting on top of the diaphragm. And the apex, if you again notice at the illustration, the apex comes out up to about the level of the clavicle. So the two lungs primarily fill the majority of the thoracic cavity in that space. So we did that, we did that. If we were to rotate the lungs and look at them from the top, they have a somewhat similar shape that we have seen before, where there are basically three surfaces that are convex, and then the inner surface is concave. So we have these two concave surfaces facing each other, and then these convex surfaces that come out. The convex surface is the part, as you can see, that is basically in contact with the wall of the thoracic cavity, which is primarily made up of the ribs, and so not surprisingly is referred to as the costal surface. And because we've seen this type of anatomy before, what do we call the indented concave region? Okay, we did. There was the lesser curvature of the stomach. That was a good one. But I was thinking more in terms of like the kidney or the lymph node when we've seen this kind. There we go. We refer to it as the hilum. So we have these hilum, or the plural, hilus, the hilum, which is the indented region. Now, there is an important point. Actually, no, I lied. We'll make that point in a second. So let me get, I can leave this here for now. And I can leave that here for now. As we know, the lungs are surrounded by a membrane. What type of membrane surrounds the lungs? A serous membrane. Excellent. And what do we call the serous membrane associated with the lungs? Pleura, absolutely, or pleural membrane. Excellent. We have one layer of that pleural membrane that is in direct contact with the surface of the lung. And what would we call that particular layer? Visceral pleura. Excellent. And we also know that this pleura lines the thoracic cavity. The part that lines the thoracic cavity, what would we call it?
Parietal. Pleura.
The parietal pleura. Excellent. But we know a third thing about these. There you go. Alex has got it. These two things are continuous with each other. So they're not separate, they're continuous. So what actually happens is our visceral pleura covers the entire costal surface, comes along the hilum, and then comes off of the lungs, where it then comes in contact with and forms the parietal. And remember also from 430, is the right and left pleural cavities continuous with each other? Are the right and left pleural cavities connected? No. Remember, each one has its own individual shrink wrap around it. So both have a visceral pleura and a parietal pleura that line the thoracic cavity, and they do not connect. Excellent. So what causes this serous membrane to come off of the, and so I have to cheat here. Let's do this. I want to grab this and I want to put it here and out like that. So I can grab that and put that here, because that hilum is that entire indented region. So let's highlight that with a brown highlighter. This whole thing is the hilum. But notice there is this region that I'll show here in yellow that doesn't have the pleura on it, where the pleura comes off. Exactly, and Alex has got it. The reason for this is where the blood vessels, this is where the blood vessels come in and out of the lungs. This is where the bronchi comes in and out of the lungs. This is where the nerves come in and out of the lungs. So this region where all of the blood vessels, all of the nerves, and the secondary bronchi connect, so let's make that a little smaller so it fits in the space. This region where these three things connect inside here is what is known as the root. Of the lungs. So inside the hilum, we have a root. And again, I've done a horrible job of drawing this, but you'll notice if we look here, great question. No, not really a lot of lymphatic vessels go to the lungs. So notice here we get this really nice view, and luckily I don't have to erase my picture to be able to show this. Notice here, so let's go there and let's go there. So again, this entire indented region is the hilum. But notice this small area in here where the visceral pleura comes off of the lungs and forms the parietal pleura, and where the arteries and the veins and the bronchi and the nerves all connect to the lungs. This region is known as the root. I want to make a point of emphasizing this because notice Your textbook takes the term hilum and puts it smack dab there in the middle. Is that part of the hilum? Absolutely. But remember, this whole thing is the hilum. This specific region in the hilum is the root. So I want to make sure we understand the difference between the hilum and the root. So remember, all pleuras, like all serous membranes, are made up of the exact same thing: a simple squamous epithelial tissue and a relar connective tissue. The simple squamous is a mesothelium. They produce a transudate. All serous membranes are the same. So really, when we talk about terms like visceral pleura and parietal pleura, or visceral pericardium and parietal pericardium, or visceral peritoneum and parietal peritoneum, all 6 of those histologically are the exact same thing. So really, if you think about it, the only difference is a location difference. If that simple squamous epithelial tissue and a relar connective tissue is on the stomach, it's a visceral peritoneum. If it's on the heart, it's a visceral pleura. If it's lining the thoracic cavity, a visceral pericardium. If it's lining the thoracic cavity, it's a parietal pleura. The only difference is their location, exactly. And not just the main difference, really the only difference. They look the same, they function the same. The only difference is their location. So all 6 of those names are really just locational names. Exact same type of membrane in 6 possible different locations. Although I guess technically you could say 8, because we do need to distinguish the right pleural from the left pleural because they are separate from each other.
So you call this root of the lung, right?
What I've highlighted in yellow would be the root of the lung. That is correct. That is the root. And the root is a small portion of the hilum. The portion where things connect and the portion where the visceral pleura comes off to form the parietal pleura. So the hilum is the indented region of the lung, and the root is where the visceral pleura comes off to become the parietal pleura, and where the blood vessels, nerves, and trachea connect. And those are your 2 definitions. All right. Questions on that? Excellent. Notice here, we can see why we have 5 secondary bronchi, because we have 5 lobes. There is a fair amount of asymmetry because, again, notice there's this big indentation in the left lung where the heart sits. So our right lung is divided up into 3 distinct lobes, and they're divided by 2 main fissures: a horizontal fissure, which primarily separates the superior from the middle lobe, and the oblique fissure, which separates the inferior lobe from the other 2. Notice on the left lobe, we just have, left lung, we just have 2 lobes and 1 fissure, an angled fissure, so an oblique fissure. Notice both have oblique fissures, but there is no horizontal fissure in our left lung, so there is no separation, but there is no middle lobe. We just have a superior and an inferior lobe in the left lung, and notice in our superior lobe, we have a cardiac notch. Excellent. Questions on that? More pretty pictures here. We see the actual cadaver. Your practice anatomy lab has some great cadaver pictures, as well as some really good models for this that you should be looking at. Your Physio X has some great, not just Physio X, I'm sorry, your interactive physiology has some great material for this. So again, remember, just because things aren't assigned, make sure you're using your resources to help you to be successful in this. All right, excellent. We already talked about our serous membranes, the visceral and the parietal pleura. In between them, of course, is the pleural cavity. And of course, that is lined with our serous fluid, which again, we call our transudate. Or because it is associated with the lungs, you will also hear the term pleural fluid as well. So both of those would be acceptable. And again, remember, is the goal to fill that pleural cavity with as much pleural fluid as we possibly can? No, of course not. We just want that thin layer of it to reduce friction in that environment. And it serves two functions. One of those functions is absolutely positively to reduce friction as it moves. However, the other important function of that pleural fluid in the pleural cavity is to produce surface tension. What do I mean by surface tension?
The lungs get increased in their volume.
True, it's going to help them to stay in context. But what is surface tension? That's absolutely correct. That's absolutely what it's going to do. It is going to allow the lungs to expand and fill with air. But what is surface tension? Can someone give me another example of surface tension? What is surface tension associated with? Come on, some of you had to have taken a general biology 300 or 400 class before getting here. Water, absolutely. Water has that mystical, magical quality to it that it is polar. And so it forms hydrogen bonds, which is why if you pour a little bit of water on your countertop, it forms a little puddle. It forms a bubble, right? It doesn't just spread out unevenly over the entire surface. It puddles together, right? It's why it hurts when you do that belly flop off the high dive into the pool because of the surface tension of the water, right? And the other thing that it does, although as cold as it's been, you probably haven't had a chance to experience this in a while, but a couple of weeks ago when it was super hot, you may be sitting out in your backyard with a nice big glass of lemonade. And that lemonade is sitting on your glass tabletop outside on your patio. And as that condensation forms on the outer surface and that glass starts sitting in water, when you go to pick up that lemonade off of your glass of lemonade off of the glass countertop of your outside patio table, what happens? Come on, none of you have experienced this? Does it slide? When two pieces of glass get water in between them, what happens? It sticks. That sticking to it is that surface tension. And as Oksana pointed out, that's exactly what we want. Remember, we're going to use our muscles to change the shape of the thoracic cavity. So as our thoracic cavity moves out, we want the lungs to stay up against it, so that when the thoracic cavity moves, the lungs move with it. That way, changing the shape of the thoracic cavity changes the shape of the lungs. And that surface tension of that transudate holding those two layers together is going to be very important. So this is going to allow us to change the shape of the lungs by changing the shape of the thoracic cavity. That ability to get it stuck to it, that surface tension in the pleural cavity is vitally important. Yeah, exactly. So notice what happens if air gets inside of that space. You have that knife wound, right? There you are sitting at the bar. Back in ancient times, we could sit at bars, arguing as you often do about who the cutest metter of one direction is. It's of course Zane. Everybody knows it's Zane, right? And so, of course, like many of these things they become quite violent, and you get that knife piercing wound between the ribs. Air gets in there, fluid gets in there, you lose that surface tension, you lose the negative pressure in there, and the lungs collapse. So now what happens is when you move your thoracic cavity, without that surface tension, without that pressure, the lung doesn't move with it. And if the lung can't move with it, you can't inflate that lung. Right? Luckily, as we know, each lung is individually shrink wrapped. So as long as one is able to inflate, it can keep you alive long enough for them to suck the air or suck the fluid out, reseal it, and form that pressure change, from that negative pressure, form that surface tension, and allow it to inflate the lungs again. All right. Here, of course, we have that pretty picture we saw way back in 430. Again, emphasizing, yep, or a collapsed lung, absolutely. When a lung collapses, it's typically because of the loss of the surface tension, the loss of the pressure here in the thoracic cavity. So when your thoracic cavity moves, the lung doesn't move with it. No, it does reduce friction as well. So it provides the surface tension and it does reduce friction because, again, it is constantly moving and your thoracic cavity is constantly sliding across the lungs, and we don't want to irritate the lungs. So it does reduce friction as well as produce that surface tension.
No, I mean, it's the only area when this transudate has this another function. For example, when we were talking about the
Oh, so yeah. Yes, we don't need that surface tension in the heart. We don't need that surface tension in the thoracic cavity. So it is just here in the lungs where that surface tension is an important part of the function of the transudate. Yes. Yep. All right. So again, here we see serous membrane, visceral, parietal. Notice this also then clearly shows our root, where the blood vessels and nerves are connecting into the entire hilum. That is our lungs, our costal surface, right and left, everything that we've talked about, we can see clearly in this nice transverse section and identify all that. And again, one other thing that I think sometimes people get confused or lost by, remember, this space that I'm highlighting right here is the pleural cavity. So is the lung actually located in the pleural cavity? No, it's actually surrounded by the pleural cavity. Right? The pleural cavity really just surrounds it. Again, very similar to what we saw in the heart. Now, with the heart, it's a pericardial sac, so you can kind of talk about it being inside. But again, these cavities really are surrounding these organs. So the lung technically isn't in the pleural cavity, well, at least not in the pleural membrane cavity, but technically it's surrounded by it. All right, excellent. We did that, we did that, we did that, we did all of that. Fun, excellent. All right, questions on that? Great, let's get back to the bronchi. As we were talking about, we have two, oops, that's not a two, primary bronchi, because we have two lungs. We have 5 primary bronchi. No, sorry, secondary bronchi. Because we have 5 lobes. Excellent. Do you think our bronchi divide further than that? Well, I spent the time to write all this, so the obvious answer is indeed yes. What would the next level of bronchi be referred to as?
Lobules?
Well, true, we'll get there, but the bronchi themselves are tertiary. Excellent. They're tertiary bronchi. Oops, got the eye there. Anyone know how many tertiary bronchi we have? Here's where things start getting a little bit trickier. Okay, many is not a bad guess, although we can actually count them. However, even in a class like this, as small as only 23 people are here right now, are we all going to have the exact same number of tertiary bronchi? It turns out not. Everybody has typically somewhere between 16 and 20 tertiary bronchi. And again, I've taken all this time to set this up. So obviously there is going to be a reason for that. And that's because everybody has somewhere between 16 and 20 additional important structures. And these additional anatomical structures are what we call bronchopulmonary segments. Bronchopulmonary refer to the pulmonary arteries and the bronchi, the tertiary bronchi. Basically, the two lungs are divided into individual compartments. The right lung 10 of these bronchopulmonary segments. I'm just going to abbreviate it BPSs here, bronchopulmonary segments. However, the left lung is where we see the variation. The left lung can have anywhere between 6 and 10 bronchopulmonary segments. Why these bronchopulmonary segments are significant is each one of these segments basically has one tertiary bronchi and one pulmonary artery that feeds into it. So if you think of this from a clinical standpoint, most, for instance, lung cancers begin in just one bronchopulmonary segment. And for instance, if you found that lung cancer early enough, well, I was still in just one bronchopulmonary segment, and that one compartment of your lung just had one airway and one blood vessel feeding into it, do you think it would be really hard to remove that one bronchopulmonary segment? No, from a clinical standpoint, it actually can be fairly easy to remove just that one segment because there's just basically one main nerve, one main artery, one main vein, one main bronchi that feed into it. So from a functional standpoint, and more importantly, from a clinical standpoint, this compartmentalization of the lungs is pretty important, pretty significant. But it does vary from person to person. All right. So I've written this. Let me go ahead and clear that and we can do it again. Primary bronchi, 2, go to the 2 lungs. So you got to remember our lungs have about the surface area of a small tennis court. So obviously losing, 1/20th of it is going to have some impact, but probably not as much as you would think. Again, there are some people who, have lung problems and get a lung transplant and get a new lung and just, basically rely on that one lung. Could you survive with just one lung? Yes. Would you necessarily be able to be efficient enough to be a professional athlete? No. But could you live a relatively normal life with just one bronchopulmonary segment or even 2 bronchopulmonary segments removed? Yeah, probably wouldn't see much of a noticeable difference in your change in lifestyle as a result of that. All right, 5 secondary that go to the 5 lobes. So the secondary bronchi are also sometimes known as the lobar bronchi. They form the tertiary bronchi, which again, there are 16 to 20 of because they feed into the bronchopulmonary segments. Your book actually has this great picture. Hold on, I'll show you those in just a second. We'll talk about it. There's this great picture, again, showing one example of this. What they've actually done is they've used they've plasticined the lungs, the airways of the lungs. And they've color coded them. So you can see the different colorations of each of the individual compartments. This allows you to see the primary bronchi. It allows you to see the secondary bronchi, and the coloration is starting at the tertiary, and then it continues to branch from there. Now, from tertiary, it goes to quaternary and so on and so forth as it continues on. In fact, there can be up to a total of 21 different levels of branching that take place as you go through them. And again, there you're going to see much more asymmetry in individuals. But here you can see how those individual compartments really do kind of separate. Now, notice also, as you look at this particular example, this person had 19. 10 as we expect in the right lung, and this individual happens to have 9 in the left. But remember, they could have as few as 6 or as many as 10. Now, obviously, as we talked about, losing one will affect lung efficiency. But one of the other questions I sometimes get is if someone who has, for instance, 6 segments on the left have more efficient or less efficient lung capacity than someone who has 10, No, the 10 ones just tend to be smaller. The 6 ones tend to be larger. But as far as I'm aware, there doesn't seem to be a significant difference in gas exchange or function associated with how many bronchopulmonary segments you have on the left side. So it seems to just be a structural difference and doesn't seem to affect anything from a functional standpoint. All right. Now, as I mentioned, after the tertiary, they're more random and elaborate in their branching, and so we don't have to worry about the naming of them. However, what we do have to talk about is what happens when you get to the deepest end of those. We'll talk about the anatomy of the bronchi in just a second, but the smallest bronchi at the very end is our bronchioles. Our bronchioles are the smallest, and one of the big differences that we'll see, one of the ways we can distinguish our bronchioles have no cartilage on their outer surface. And they have a simple cuboidal epithelial tissue. All right. So here we see a nice illustration that does a good job of showing this. And actually, this is a good stopping point. Let's actually go ahead and take our second break here. And then we have a little bit more still to talk about, but I think this is a good natural stopping point for now. So let's go ahead and we'll finish the discussion of the anatomy of this, then that transition. I think this was one of the questions that was asked before. So let's save this for after the break. So again, it is 2:37. So it means that we will come back at, what is that, 2:52? That sounds like 15 minutes, right? And I will start the recording at that point. All right, so any questions before we take our next break? All right, see you in 15 minutes. Any questions before we move on? All right, so as we've talked about, we understand what is going on in our, where's my pointer? Here in our primary, here in our secondary, here in our tertiary branches of this. And we've talked about why the numbers that we have. And as we talked about, there can be a further order, as many as 21 different orders of branching that can take place. But aside from the number and obviously the size getting smaller of this, there's a transition in the anatomy of this as well. In the primary bronchi, like in the trachea, we have C-shaped cartilage rings on the outside. And inside, it is lined with a ciliated pseudostratified columnar in epithelial tissue. Just like the trachea. So just like the trachea, we have this anatomy to it. And not surprisingly, again, it's going to have a muscular wall, but a relatively thin, smooth muscle layer. Because after all, what would the function of our smooth muscle layer be in this, in a circular organ like this?
Change in diameter.
Change in diameter. And if you have a big, huge circular ring of cartilage, right, or in this case, a C, right, you can have minimal change, but you're not going to be able to make big changes in it. So with that C-shaped cartilage, we can make minor changes to the size, but we're not going to be able to have a huge effect on it. But notice, as it transitions into the secondary and the tertiary, notice we are going to get some changes. First, on the outside, the C-shaped rings break up into irregular plates of cartilage. On the inside, oops, if I spell inside right, we transition to a simple columnar epithelium, and we get a thicker, smooth muscle layer. Now with irregular plates, we don't have as much restriction. And so we're able to have a bigger effect on the diameter and changing the diameter of the bronchi at this point. So we are going to get more smooth muscle for more change that's going to be able to take place because we have those irregular plates and it transitions. So again, it's not exactly at the border of first to second, but as we're transitioning from those second into those third branches, those tertiary branches, by the time you reach the tertiary branches, the epithelium has become simple columnar. So it's always ciliated pseudostratified in the primary. It's always simple columnar in the tertiary, and it kind of transitions through the second. All right. Notice our bronchi, as they continue to get smaller and smaller, they still have small irregular plates of cartilage. They still are going to be lined with that simple columnar. But remember, as you see here, when we get to the level of a bronchiole, the bronchioles, notice, have no cartilage on the outside. They're still going to have smooth muscle, a good smooth muscle wall to them, but they on the inside now have simple cuboidal epithelium. These bronchioles eventually lead into the alveoli, and obviously they have no cartilage, but remember our alveoli is just a single layer of simple squamous epithelium. Someone, I think it was Oksana, but I don't remember exactly who, I've certain, asked about those transition points of the epithelial tissues in the bronchi. And now we can kind of get an understanding of how and where that is going to occur. All right.
How it affects the function?
I'm sorry?
How it affects the function, this changing?
It's a great question. Well, so obviously the primary bronchi and the trachea are still able to capture dust and debris with their cilia and move it up and away. But by the time we get into the lungs, which is where the secondary and the tertiary bronchi are, there's no more cilia. So anything that gets in there, unfortunately, is going to be trapped in there. Now we have macrophages and other things that eventually can hopefully break them down and move them out, but they're not going to be able to move them that way. But as far as the simple cuboidal versus simple columnar, they're still just passageways. So really they're not, there's no absorption, there's no secretion or anything like that taking place. So my guess is that it probably has more to do with the size. As it gets smaller and smaller, we transition from columnar to cuboidal. Until we get to the alveoli, where there the size definitely matters. Being the simple squamous there makes the difference because that's where the gas exchange is going to take place.
Okay, thank you.
But yeah, the simple cuboidal and simple columnar, there's really no difference in their function. My guess is, like I said, it probably has more to do with size. All right, so hopefully that makes some semblance of sense. So again, we're going to get a change in the cartilage. We're going to get a change in the epithelium, and we're going to get a change in the amount of smooth muscle as we go from primary all the way down to the bronchioles. All right, now, again, we start primary bronchi, secondary bronchi, tertiary bronchi, and then the bronchi continue to get smaller and smaller and smaller until we reach the bronchioles. At the very end of our conducting zone, and remember our conducting zone is just about moving and processing the air, we reach what is known as the terminal bronchiole. That terminal bronchiole is the very last level before we reach the respiratory zone. So it has one job and one job only, moving the air. Notice again, we see the smooth muscle on the outer surface. It's lined with a simple cuboidal epithelial tissue. And again, notice there is no cartilage. Notice that a terminal bronchiole will feed into a small compartment of the lungs known as a pulmonary lobule. So this clustered structure we have here is a pulmonary lobule. And that pulmonary lobule is fed into by a respiratory bronchiole. And here's the difference. Let me go ahead. I'm going to cheat and just draw this because it's super simple. Bronchiole. Smooth muscle, simple columnar epithelial tissue. Bronchiole, smooth muscle, simple columnar epithelial tissue. The only difference between a terminal bronchiole and a respiratory bronchiole is one thing and one thing only. A respiratory bronchiole will have an alveolus on it. One or more alveoli on them. Alveoli, of course, are where gas exchange takes place. So if you have a bronchiole that has one alveolus on it, that bronchiole can participate in gas exchange. Is it going to be the primary location where gas exchange takes place? No, of course not. One alveolus isn't what we're going to rely on to get all the oxygen that we need for our body. But technically, if you have a an alveolus on you, you can, to some extent, participate in gas exchange. Whereas if you don't have alveoli on your coming off of you, then you can't participate in gas exchange. And it's really just that simple. If we look back at the illustration, we can see right here that this bronchiole has an alveolus on it, has two alveolus on it. And so as a result of it, it can play a limited role in our gas exchange, whereas this one up here and this one up here don't. So these would be terminal bronchioles, and this would be a respiratory bronchiole. And that's really going to be the only difference. This is significant when we look at this histologically. I'm going to cheat and jump ahead. Notice on our illustration here, they show us that this is a respiratory bronchiole, right? We can see on this that it is a bronchiole because we can actually see that there's a little bit of smooth muscle. If we were a little bit closer, we'd be able to see that this is lined with a simple cuboidal epithelial tissue. But how do they know this is a respiratory bronchiole? What does a respiratory bronchiole have to have? What does a respiratory bronchiole have to be able to do? The microphone is on, right? Yeah,
Alveoli, right?
exactly. And look, lo and behold, this little ball-shaped structure sticking off of it is an alveolus. It looks like there's a tiny bit of an alveolus right here, although I wouldn't use that on an example, but this one here is a very nice example of one. We see a very clear example of an alveolus coming off of this bronchi, so it is clearly a respiratory bronchiole because it has an alveolus on it. All right, it really is just that simple. All righty, so those respiratory bronchioles then feed into those lobules that we talked about, and that is where the primary alveoli are located, and that is where most of the gas exchange takes place. But everything from the respiratory bronchioles down is considered part of the respiratory zone, the part of the respiratory system where external gas exchange takes place. So I guess we should be a little bit more specific by saying that we are talking about external gas exchange taking place here. All right. Questions on that? Excellent. Our lungs are basically big bags of air in each lung. And again, I'm not the one who counted this, but so I'll take their word for them. There's about 300 million alveoli per lung. So not surprisingly, when we look at it under the microscope, it is mostly just air-filled space lined with those simple squamous epithelial tissues. Notice our alveoli are not just randomly organized though. If we cheat and go back to the previous picture, we can see that alveoli are located in clusters. These clusters are what we call alveolar sacs. Kind of like grapes are found in a cluster. You have a cluster of grapes. And does hope hold the grapes together when you have a cluster of grapes? If you are like me, you like to have your significant other just hold that big bunch of grapes over your head where you can occasionally bite one off of them. What's holding those grapes together? Is it hope? Dreams? The dreams and hopes of the grapes that keep them together? No, you may not see it from the outside, but you know there is a stems underneath, and those stems connect underneath to hold them together. And that's exactly what happens here. Notice on this illustration, we can see there is an alveolar duct, which is basically the stem that all the alveoli connect to. And we see it in the histology slide as well. The other example I like to use for what an alveolar duct is like the hallway at a hotel. Right, you get your hotel key, you go up to the 14th floor, and as you get off on the 14th floor, there's a big long hallway where all the rooms are off to the side, and all the doors come off that main hallway. Well, that's basically what our alveolar duct is. It is just a big hallway. And notice when you look at it histologically, you really identify it more by the void. This space, this void, is that hallway that all the rooms, all the alveoli come off of. So we have all these alveoli coming off of that main hallway. And so we've got this big cluster of grapes. We have this big row of rooms that are the alveoli coming off of that alveolar duct. So notice the alveolar duct doesn't have a lot of structure to it. To be the same thing in the hotel. It would be very hard to rip a hallway out of a hotel. It's really more just the small space that connects all the rooms together. And that's what the alveolar duct is here. It is just that small space that holds all the alveoli together. And so it mostly makes up most of this lung's volume. It's a very light, airy, collapsible tissue as a result of that. And like I said, it has a huge surface area. If you were to take the lung surface area out and spread it over the surface of the floor, it would hurt a lot. All right. As I mentioned here, we see another nice illustration with this anatomy that we've been talking about. Here's our terminal bronchiole, that last bronchiole before the end of the conducting zone where it does nothing but move and process the air. Then we have our respiratory bronchioles that have a couple alveoli on them. They feed into our alveolar sac, and that alveolar sac is made of the singular duct with all the cluster of alveoli wrapped around it. So we did all of that. Excellent and excellent. All right. Questions on this gross anatomy? All right, stunned silence, love that. Let's talk about the microscopic anatomy. If we cut into one of those alveolar sacs, what we see are a bunch of alveoli, and we get a better sense of the anatomy. Notice over here in red, we have a colorized electron microscopy view that shows the pulmonary capillaries that wrap around the individual alveoli. And here, if we were to take a cross section through it, would be essentially what it would look like. Here we see a cross section through a single alveolus. Notice, like the illustration shows, it is surrounded by pulmonary capillaries. And remember, all capillaries are so small that our red blood cells have to line up single file to be able to pass through them. So they're tiny in relation to this big, huge air-filled sac that they surround. Now, this alveolus, as you can see, is made up of or contains several different types of cells. The primary cells that make up the alveolus are what are known as the type 1 pneumocytes. That's these beige cells. They're the simple squamous cells that form the walls, or most of the walls of the alveolus. However, notice within the walls of the alveolus, there are these occasional green cells. Now, of course, they're not green in real life, but they're here to represent a second type of pneumocyte, what is called a type 2 pneumocyte. So when you identify them on the exam, you can certainly say type 2 pneumocyte, because again, that is incredibly fun to say and spell. But they do have another name, and that other name is septal cells. And not surprisingly, these septal cells have a specific function. Their function is to produce a special fluid called surfactant. Surfactant has one job and pretty much one job only. Its job is to break up the surface tension of the water inside the alveolus. How many people here have blown up a balloon before? Anyone? No one's blown up a balloon. Excellent. One person has. Excellent. Oh, there we go. Better. What's the very first thing you do before you blow up a balloon? Okay.
Take a deep breath in.
Take a deep breath in. Excellent. I like Nick's answer the best, though. You take that balloon and you stretch it out.
Oh,
Well,
yeah.
why do you stretch it out? So that the elasticity of it is warmed up, so it makes it easier for it to fill up without exploding. Is that why you stretch it out?
You stretch it out.
I don't know, but everybody does it, right? You always do it. Nobody knows why. There you go. Alex has got it. You do it to separate the two sides. The two sides, while it's stuck in the pack, are stuck together. And when those two sides are stuck together, how easy is it to get air into there to separate those two sides and fill up the balloon? Is that really easy to do? No. But if you stretch it out, that separates the sides a little bit. And once the sides are separated, it's much, much easier to blow that balloon up. That's kind of how surface tension would work. Remember, we've used all this mucous membrane to make this big, huge, moist air that is getting down to the alveolus. So this alveolus is covered with moist air. It's covered with water. And as we talked about out on the lungs, two surfaces that have water on them will stick together thanks to that surface tension. Now, that's a good thing on the outside of the lung, because when our thoracic cavity moves, our lungs move with it. But every time we fill our lungs, do we want to have to force the alveolus open if those two sides of it stuck together? No, that'd be really hard to do. So what the surfactant in here does is here on the inside of the lungs, surface tension is bad. So surface tension good outside the lungs. But surface tension is bad inside the lungs. And so what that surfactant does is breaks up the surface tension inside the alveolus so that the alveoli don't collapse. Because if the alveoli collapses, it's going to be much harder to inflate it. And if we can't get air into it, we can't get gas exchange to take place. Again, we talked about grandpa having to actively breathe using inhale and outhale. He worked for 40 years, 30 of them in the coal mines, inhaling all that dust, causing all that damage. And one of the things that happen as we age is our lungs produce less surfactant. So when grandpa takes a breath in, some of his alveoli are damaged. Some of his alveoli don't stretch out as much, right? The lungs have lost their elasticity, and some of them have collapsed because of a loss of surfactant. So every breath that grandpa takes isn't going to be as effective at getting gas exchange to take place. So typically what happens to our respiration rate as we age? From adulthood towards elderly?
Decreases.
It increases. We have to do more. Now be careful. From infant to adult, definitely there's a decrease in the rate of breathing. So infants breathe at a mutantly fast rate because they have tiny little lungs. So from an infant to adulthood, there's a decrease. But from adulthood to old age, the number of breaths grandpa has to take goes up because each breath he inhales is not as efficient at getting the gas exchange to take place. And so as a result of that, he has to take more breaths a minute than you do to be able to meet the needs of his body, which further exhausts him. Right? Just being old is exhausting. And I know from experience. Excellent. Notice there are some additional cells in here, these blue cells. These blue cells are not actually part of the alveoli, but migrate through the alveoli. And these are actually what are known as alveolar macrophages. Right? They're also commonly referred to as dust cells, and I'm okay with you using that term. But as an alveolar macrophage, that probably can give you an indication of what their function might be. What would macrophages do?
Eat phagocytes.
Yeah, absolutely. So if there are any harmful pathogens or any other type of irritants that made it down into the alveolus, these alveolar macrophages can help to destroy them. Right, so they can help to play a role in limiting those and therefore limiting their ability to get into our body. All right. Now, obviously, the other thing that we have down here, as we mentioned, is this is where our external gas exchange takes place. This is where we are going to have our respiratory membrane, or what we also can think of as our air-blood barrier. Right? This is where the stuff in the air gets into our blood and vice versa. This is where our gas exchange is going to take place. And one of the reasons we need it so deeply embedded deep inside of our chests, past all these funky and specialized airways, is because it is so incredibly thin, we need to limit any kind of dust and debris and irritants and pathogens that can get to it. Because as we know now, our alveolar wall is made up of what type of tissue?
Simple squamous.
Simple squamous epithelial tissue. Our capillaries are made up of what type of tissue? Simple squamous epithelial tissue. Basically, we have two simple squamous epithelial tissues with their basement membranes holding them together. That's it. We have the epithelial tissue of the alveolus. We have the endothelium of our pulmonary capillary, and we have their fused basement membranes, their fused basal laminae, and that's it. Your respiratory membrane is tiny. It's only about a ½ micron in diameter. So again, you have the surface area of a small tennis court and a thickness of ½ micron. No ATP is used. We have a massive surface area, short distance it has to travel, warm, moist air, so that that passive diffusion of gases can occur as efficiently as possible. Questions on that? All right. There is one last piece of anatomy we need to talk about. So let's do this first. I want to check something real quickly. Would have been very efficient if I had bothered to do this stuff ahead of time, but that would have required a level of efficiency that apparently I lack.
I have a quick question.
I have a quick answer.
What happens to the alveoli that collapsed, that they are not open anymore?
Well, one of the problems with that is that we're not going to be able to inflate the lungs anymore. So that is going to be an issue. You can reduce it. Now, again, in time, can you produce enough surfactant to be able to relax it or open it? Can a forceful breathing help to get it open and things along those lines? Yeah, it can, but in some cases it can become limited. It can become damaged as a result of that. So it is something that can be tricky and challenging. Oh, let's look at this.
What happened to them? I mean, to the alveoli, what happened to them? Are like, they are dying, their cells are dying or something like this?
Yeah. So yes, that could happen. If they're not getting the oxygen that they need and the gas exchange isn't taking place, then those alveoli could potentially die, yes.
Okay.
All right. Before we go on to this last thing, let's go ahead and take a look, because I think this is going to show me what I want. I'm hoping. I guess I should actually look at it ahead of time. Yeah, this is what I wanted. That's what I want. Yeah. Perfect. Excellent. All right. So let's go all the way back. For those of you who have not doing this already, first and foremost, shame on you. Perfect. Excellent. No. So, as you know, under the study tools, one of the study tools that is here is our virtual anatomy lab. This virtual anatomy lab is, oh no, that's not the one I wanted. Sorry. The CRC virtual anatomy lab. The other one's good too, but I didn't look at that one. I wanted to look at this one. Virtual anatomy lab. Here in the virtual anatomy lab, we can go to the respiratory system and it shows us the model and charts. Shows us many of the things we've already been talking about. And again, so these have labeled and unlabeled illustrations to them, which are really great. Another great view. And again, you can see those 4 bumps of the corniculate and cuneiform cartilages on there from that view. We've talked about a lot of these things we've seen. Here we see some of the models that we have of the larynx. So again, this one happens to have the hyoid held in place and some of the things associated with that. Here we see the arytenoid and corniculate cartilages. This model actually you can pull and it rotates and opens it. So it's kind of fun that way. Lets you play with this. Here we have these nice bronchial trees that have a larynx on top of them. And again, great job of labeling these. This is the model that I wanted to show you because I love this model. And of course, if I love a model, that should definitely be a guarantee that it's going to be on the exam. It's also going to lead very much into what we have to talk about last for today. And it does an almost perfect job of showing us the pathways of the anatomy of this. But let's start with this first, in that it does a good job of showing us a lobule. We have our terminal bronchi, which come into a respiratory bronchiole that come into the alveolar ducts that feed into the alveoli. We see our blood supply to them and everything that's to this as well. So this is an excellent model that does almost everything perfect except for one huge major error that this makes. And I'm curious, yeah, that's what I was curious if they did that. All right, excellent. So we have this nice model. So again, lots of good pictures of lots of models that do a good job of showing the things, the chart. All these things are excellent. Really, really great that they have here. I want to leave it at this picture here for a second so that we can talk about the last thing I wanted to talk about in the lecture. And that is that there are not one, but two blood supplies to the lungs. I know based on what we talked about during the cardiovascular system, that may be a tiny bit surprising, but let's think about what it is. When we think of blood supply to the lungs, what blood supply do we think of?
Pulmonary vein and arteries.
Right, the pulmonary circulation. Excellent. As we know, that pulmonary circulation's job is what? What is the job of the pulmonary circulation?
To take poor oxygen poor blood and return to the heart oxygen-rich blood.
Excellent. The job is to take the blood to the alveoli, or more specifically to the pulmonary capillaries for gas exchange. Excellent. And conveniently enough, because we already did the cardiovascular system, we could draw this pathway if we wanted. It's been a very long time since I've drawn a heart. Let's see if I'm still able to do it very efficiently. Yep. Lo and behold, there is my heart. 1, 4 chambers. Excellent. Someone remind me again, where does the pulmonary circulation begin? Let's hold on. I moved the chat out of the way, so I'm going to bring it up. Excellent. So it starts in the right ventricle. Excellent. And from the right ventricle, we've got what that comes out of the right ventricle.
Oxygen poor blood.
Excellent. So we have oxygen poor blood that comes out. This is too big. Comes out of the right ventricle into the pulmonary trunk. That pulmonary trunk then, of course, does what? Like all trunks, it
Branches.
branches almost immediately to produce what? Right and left, pulmonary.
Yeah.
And those pulmonary arteries ultimately lead to the pulmonary capillaries. Excellent. So what we need over here is a lung. Here's my lung. And we know inside the lung, we've got those bronchi that are coming in, primary, secondary bronchi, branching into tertiary bronchi, and so on and so forth. And obviously, this is not being drawn to scale, but we know inside of here is an alveolus. So what we know is that our oxygen poor blood leaves the right ventricle out. And again, we goes wrap around, but for simplicity's sake, it's going to come in here, go to the root. In the root, it's going to come in, and it actually follows the bronchi down to the alveoli, where it then forms our pulmonary capillaries. So this right here is our pulmonary capillary. Excellent. where external gas exchange takes place. What gases are exchanged? I already wrote them, so tell me. Oxygen and CO2. Remind me the direction oxygen goes again.
Into.
Again, into doesn't tell me enough information. I need more than that. I need to know what it's going into. Excellent. I like that.
Capillary.
Excellent. All right. So CO2, oxygen is going into the blood, but let's make a sentence out of it. Out of the air into the blood, right? Which way does CO2 move?
Out of blood to the air.
Into the air. Excellent. Excellent. We now have oxygen-rich blood. There. 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 And that oxygen-rich blood. And notice, oops. Now that we have our oxygen-rich blood, do I really need to worry about following the bronchi back anymore? No, I can chart my own path and come out here and eventually leave out the root. And where am I going to go?
To the left atrium.
Yeah, and actually, you know what? I'm going to totally cheat on this. Excellent. So I'm going to, even though it's not the root, again, because of the simplicity of the schematic of this, I'm going to come out large because that's what I like to do. And it comes back into the left atrium. So this is our pulmonary vein. And again, how many pulmonary veins do we have?
4.
4. Excellent. 4 pulmonary veins into the left atrium. Excellent. And of course, at this point, our blood is oxygen rich. Excellent. We all comfortable with that? Really no new information here at all, right? Yes?
Yes, it's come.
Excellent. But it turns out there is a second circulation to the lungs that we didn't talk about. And this second circulation to the lungs is what we call the bronchial circulation. Now, alveoli have that oxygen, have that CO2, have that gas exchange takes place. Oh, that's crazy big. We don't need that. I want that, and I want, let's do that. Bingo. Gas exchange that is taking place in here. Oxygen and nutrients, oxygen and CO2 changing positions, yammer, yammer, yammer. Everything that goes along with that. And so obviously, while oxygen is passing through it, it could get the oxygen that it needs by simple diffusion. It can get rid of the CO2 it needs by simple diffusion. And that pulmonary circulation might have nutrients or chemical signals or other things along that way that'll help it as well. And that's great. But remember, the alveoli is the only place where gas exchange takes place. So in that big primary or secondary or even tertiary bronchi, is it going to be able to get all the oxygen and all the nutrients that it needs just from the air that's passing through it? No. So what happens is basically every other part of the lungs, with the exception of the alveoli, need a blood supply. Heck, the heart itself needed its own blood supply, right? And it's got blood inside of it. So it's not surprising that the same thing is true for the lungs. So basically the function of the bronchial circulation is to take blood to all parts of the lungs except the alveoli. Right? And the reason for this isn't going to be for gas exchange, but instead to provide the oxygen, the nutrients, and the waste removal. That kind of process where you would have a oxygen-rich artery carrying blood to a location to provide oxygen, provide nutrients, pick up waste, and carry that oxygen-poor blood back in a vein. Doesn't that sound an awful lot like a systemic circulation instead of the pulmonary circulation? Well, it should, because that's exactly what this is. This is actually a part of the systemic circulation. And that should help us a lot, because we also know how the systemic circulation works. Where does our systemic circulation begin?
The heart, from the heart.
Which side of the heart? There we go, left ventricle. Begins in the left ventricle. In the left ventricle, we have oxygen-rich, whoops. Actually, let's do that. I like that. Coming out of our, no, do that. Excellent. Coming out of our left ventricle into what blood vessel?
Aorta.
Excellent. So into the ascending aorta. From there, where would we go?
Aorta.
Aortic arch. From there, we would go into the true descending, but we can probably be a little more specific than descending aorta. Thoracic. Excellent. And our bronchial arteries come off of the thoracic aorta. And these bronchial arteries' job is to carry blood. And again, they're also going to need to enter into the root like everything else does. And their job is to provide blood to a systemic capillary. Where am I going to put this? This isn't good. I'll have to put it over here. Provides blood to a systemic capillary. And let's go ahead and put our double arrow here again. The systemic capillary, for instance, that's taking place right here. And in the systemic capillary, oops, that didn't work. There. Uh-oh, no. There we go. Perfect. There. All right. So, brings blood to a systemic capillary in the lungs. And in that, gas exchange is going to take place. But what kind of gas exchange would this be? Internal or external? Internal gas exchange takes place. So our internal gas exchange takes place. Of course, it's still oxygen and CO2 that are moving in these cases, but here in the systemic artery, which way is oxygen moving? Excellent. Okay, so but we want to be careful with the to the lungs. We want to be careful with that because to the lungs could be interpreted to the alveolus. So excellent. I like this. So in this case, again, let's put it all together. Out of the blood and into, and since we use blood, we want to go with, so instead of blood and tissue, we want to go medium to medium. So it'd go out of the blood into the What's the fluid of the tissue? Tissue is not wrong, but it's not medium to medium. We could say systemic capillary into the tissue, interstitial fluid. Excellent. Out of the blood into the interstitial fluid for the tissue. And which way does CO2 move?
Out interstitial fluid.
Out of the interstitial fluid into the blood. Excellent. At which case, of course, oops, yeah, our blood is now oxygen poor. And as it leaves, where does this oxygen poor blood need to go? Back to the right atrium. But you know what? That's far. I don't want to go all the way to the right atrium. And there's a perfectly good blood vessel right here above me that's going back to the heart anyway. So why don't I just hop a ride? As it turns out, the bronchial veins do not go all the way back to the heart. Instead, they feed their oxygen poor blood into the pulmonary veins. And so blood from both circulations all go back by just one route back to the heart. Notice, as we mentioned, this blood, when it comes back to the left atrium, we know is oxygen rich. Well, guess what? Here where the gas exchange took place, it was, and I think the technical term for it is really, really oxygen rich. I'll make that red. And by the time it reaches back to the heart, it is slightly less. Than it was when it first took place in gas exchange. And are we going to need to know the actual numbers of how that change takes place? Yes, but not today. So those are some of the important numbers we will talk about. So as we'll see, when gas exchange takes place, how much oxygen gets into the blood in the capillary there, it's going to be slightly less when it gets back to the heart, because of those damn lazy bronchial veins. They can't carry their blood back to the heart on their own, so instead, they dump it into the pulmonary veins that are on their way back. So notice, basically, 2 circulations go to the lungs, but only 1 circulation comes back.
I'm a little confused. May I ask you?
Yep, absolutely.
So pulmonary veins go into the lungs veins go to the pulmonary veins, which are oxygen rich, right?
Yep. The bronchial veins feed into the pulmonary veins. That is correct. So they are dumping some oxygen poor blood into that oxygen-rich pulmonary vein. Think of it this way. You were talking before about that hot cup of coffee or that hot soup. You make that really, really hot soup, and you realize it's too hot to drink. So as you mentioned, one thing you could do is you could blow on it. The other thing you could do is put an ice cube in it. If you put an ice cube in it, you're putting really cold water into really hot water. And when you do that, does it freeze your bowl of soup?
No.
No, it just drops the temperature a little bit. It's still going to be hot. It's just a little less hot. And that's what's happening here. We're dumping a small amount of oxygen poor blood into those pulmonary veins. So is it going to be as perfectly oxygenated as it was before? No. But is it still going to be very oxygenated? Yes.
I mean, still enough, right?
Yeah. In fact, again, you don't need to know this number yet, but next week we'll learn it. So I'll just give it to you. Here, where the gas exchange takes place, the partial pressure of oxygen. The partial pressure of oxygen here, where gas exchange takes place in that pulmonary capillary, it is 104 millimeters of mercury. When it gets back to the heart, it is 100 millimeters of mercury.
Not significant change.
Not a big change. Still very, very oxygen rich.
Okay. Okay, thank you.
Yep. So it's a change, but it's that drop of ice into your soup. It's still going to be pretty hot. All right. Excellent. So two things I want to mention here. So again, we have these two circulations. We talked about that. We drew all of that. We discussed that. Our pulmonary arteries, our pulmonary veins, the condition of the blood, all the things that go along with that. And we talked about our bronchial arteries and our bronchial veins and those differences there. All right. I mention all of this because I want to come back to this model. And I want to do one quick thing. There. If we look at this model now is perfect. Notice we see some blood vessels. Here is a blood vessel traveling along the alveolar ducts, coming to the alveoli. And what color is this one?
Is it gray?
It's gray, but what's it supposed to be? It's just gray because it's a bad picture. What color do you think it is? It's blue. Why is it blue? What does that blue represent for us?
Oxygen, poor blood.
Yep, and this oxygen poor blood, notice, is going to a capillary where suddenly the blood becomes red. So what blood vessel, and let's go ahead and label this. What blood vessel do you think this is right here? Branch of what blood vessel? Pulmonary artery, excellent. Perfect. And as we can see, it feeds blood into a capillary. No, this can't be seen very well, can it? Let's change the color of that. There we go. There we go. Excellent. What kind of capillary wrapped around the alveolus do you think that that is? Pulmonary capillary, excellent. And in that pulmonary capillary, what type of gas exchange is taking place? External gas exchange. Which way does oxygen move in external gas exchange? From the air into the blood, excellent. Which way does CO2 move? Excellent. Notice then this blood from these capillaries feed into these red, dark red blood vessels that no longer have to follow the alveoli and can work their way back. So, oh, here's a good spot for it. This dark red blood vessel, there you go, look at that. I didn't even have to answer the question. Is indeed an example of the pulmonary vein, a branch of the pulmonary vein. However, as I look closely at this model, I see that there happen to also be some light red colored blood vessels here as well. I noticed these light red colored blood vessels happen to be in the connective tissue septum of my lobule and don't seem to go anywhere near the alveoli. What might this lighter red blood vessel be?
Bronchial vein.
A bronchial vein, excellent. Now, two things I want you to be careful about. Bronchial, referring to the bronchi, not bronchiole. Remember, those are the tiny bronchi. So it is bronchiole, not bronchiole, bronchial. And again, this is a systemic blood vessel. So they are red and they're arteries. So this is the bronchial artery. Notice this bronchial artery happens to bring blood to a capillary that is not on an alveolus. What type of capillary might this be?
Now it's bronchial veins.
Well, true, but that makes it a systemic capillary. Excellent. And of course, as we know, in a systemic capillary, which way does oxygen move? From the blood into the interstitial fluid. Excellent. Well, careful. Moves from the blood out of the systemic capillary into the interstitial fluid. And then CO2, of course, moves from the interstitial fluid into the blood. Notice lastly, there is going to be, oh, and here's the best example of it. Then we have one of those veins that happens to feed into our dark red blood vessel. So what is this vein that is feeding into the dark red blood vessel? There's our bronchial vein, exactly. Too lazy to take the blood back to the heart on its own, so it just dumps it into the pulmonary vein and lets the pulmonary vein do the work. Notice, as I mentioned here, if we just look at the model this way, it's perfect. It does an amazing job of showing us all of our blood vessels, the anatomy of the lobule, and all those great things. But notice I had to cut the top off of it. So any questions on this before I show you the fatal flaw in this model? All right, excellent. I'm gonna have to cheat a little bit here. I gotta move all this stuff to the side. So that goes there. That's gonna go there. And that one, I'm just gonna delete. Excellent. This I'll move up here. I think that's enough out of the way. That I go there. Oops. I don't know what I just did there. Let's go ahead and delete that. Delete that. Move that there. Move that there. Now, notice, as we mentioned, here in blue is our pulmonary artery. And remember, as we mentioned, the pulmonary artery is supposed to follow the bronchi down. Here in red is what is clearly supposed to represent the pulmonary vein, and it doesn't follow the bronchi back. But what they did, I'm sure our artists did when they were making the top of this model is, oh, this is supposed to be an artery, it must be red. This is a vein, it must be blue. So notice these two blood vessels are not where they're supposed to be. The vein in red should be separate from the alveoli, separate from the, not alveoli, separate from the bronchi, whereas the artery should be on the bronchi. So they have these two in the wrong position. But like I said, as long as you cut off the top and ignore the top of this, right? If we do that, then it's perfect. All right. So I don't want you to get confused when you look at this model by itself. No, as long as you cut up the top, it will show you perfectly what you need to see. All right. Questions on that? So mentioned how much I love this. I spent a tremendous amount of time on it. Should you be surprised to see this on the lab exam? No, in fact, I'd be surprised if it wasn't on the lab exam. Of course, it is randomly assigned, so who knows, maybe you won't get it, but I guarantee you there are pictures of it in the pool for the lab exam. So let's say it that way. Who knows whether you'll see them or not. All right, excellent. With that, that is pretty much the extent of the anatomy we need to know. So what we will do on Tuesday, after everybody votes, is we will come back and start talking about the physiology, first talking about ventilation and everything else associated with that. All right. Questions on any of this? I'm finishing a bit early today, but so I'm happy to answer any questions, or if there's any other thing about the anatomy, microscopic or gross anatomy that you have any questions on, I'm happy to answer those questions. But that is all I have for you today. So we were able to cover all the anatomy. So now we can spend the basically next week talking all physiology all the time. I see people fleeing from the building already, so I guess I'll ask one more time. Any last questions? All right, be safe, be careful, listen to the doctors, do not listen to the politicians, vote, make your voice heard, all right? Be part of how you want to shape this world. So please do that, make wise choices this weekend, be safe, have fun, study hard, and I will see you guys on Tuesday.